Good afternoon, good evening, good morning, and a warm welcome from Rutgers, the State University of New Jersey, and Princeton University to all of you joining us from around the world. Today, the Rutgers Institute of Quantitative Biomedicine crash course was co-organized with the Rutgers Cancer Institute of New Jersey and Princeton University. My name is Stephen Burley. As University Professor and Henry Rutgers Chair, I serve as founding director of the Institute for Quantitative Biomedicine. Could you please suppress that? I serve as founding director of the Institute for Quantitative Biomedicine, director of the RCSB Protein Data Bank, and Cancer Pharmacology Program co-lead at the Cancer Institute. And I'm also a member of the Department of Chemistry and Chemical Biology. Our topic for today's crash course is intrinsically disordered biological macromolecules in cellular signaling and regulation. Before I introduce the Institute, I need to thank my co-organizers, Dr. Edmund Latimer, Associate Director for Research and Education Affairs at CINJ, and Professor of Surgery, Biochemistry, and Molecular Biology at Robert Wood Johnson Medical School. Dr. Clifford Brangwyn is Director of the Princeton Bioengineering Initiative, June K. Wu Class of '92 Professor in Engineering, and Professor of Chemical and Biological Engineering. He is also a full investigator in the Howard Hughes Medical Institute. The Institute for Quantitative Biomedicine was established in 2014 under the umbrella of past President Barchi's strategic plan as a center of excellence for interdisciplinary research and education, operating at the interface between the quantitative and the biomedical sciences. It is home of the RCSB Protein Data Bank, the US Data Center of the Worldwide Protein Data Bank, resident faculty members working in structural biology, computational chemistry, molecular biophysics, and statistics, the Quantitative Biomedicine Graduate Program, and various university core facilities supporting cryo-electron microscopy, mass spectrometry, and nuclear magnetic resonance spectroscopy, all for biological macromolecules. Dr. Steve Labuti, director of the Rutgers Cancer Institute of New Jersey, has kindly agreed to say a few words of welcome and introduction on behalf of CINJ.
Thank you very much, Steve, for that introduction. And it's a real pleasure to join all of you this afternoon, this morning, and this evening, depending on what time zone you're in. The Cancer Institute in New Jersey and the Institute of Quantitative Biomedicine have a long-standing partnership. Over the last 8 years since IQB was founded, we have worked together to advance the areas of overlap between IQB's mission in research and discovery and CINJ's mission in focusing on progress and discovery in cancer. Princeton University and Rutgers University are consortia partners for the Cancer Institute. And together, CINJ, IQB, and our two university parents, Rutgers and Princeton, work together very collaboratively to advance important areas of mutual scientific discovery. This is the 4th annual joint conference hosted by CINJ and IQB, and I'm sure this exchange of information and the great presentations that you'll all be hearing will help to further and foster collaborations and new discoveries, hopefully leading to a better understanding of disease and novel therapeutics. And so on behalf of the faculty and members of the Cancer Institute in New Jersey, I thank Steve Burley for his leadership of IQB and thank him for the opportunity to address you all today and to welcome you to this conference.
Thank you very much, Steve. My colleagues and I are deeply grateful for your support of the Institute for Quantitative Biomedicine and the RCSB Protein Data Bank. My co-organizer, Professor Ned Latimer, will now summarize the course objectives.
Jen, you're ignoring one important aspect of the loan. They can see me.
Thank you, Stephen, and good afternoon. From when we began to put pen to paper for this conference in the fall, there's been a great deal of excitement across our institutions for the subject, We're particularly pleased at the ability to recruit such an outstanding group of speakers today. Enthusiasm is also validated by the rapidly increasing number of participants. We understand is going to reach upwards to 600 by the end of the conference. Living cells contain distinct subcompartments such as secretory vesicles and endoplasmic reticulum, which facilitate spatiotemporal regulation of biological processes and biochemical reactions. In addition to these well-known membrane encapsulated organelles, there are many intracellular organelles not enclosed by membranes that retain coherent structures capable of compartmentalizing and concentrating well-defined cadres of biomolecules. These structures play diverse roles in fundamental biology and are also increasingly being implicated in protein aggregation diseases. Studies have shown that such membrane-less assemblies exhibit liquid-like features, and liquid phase condensation is now thought to represent a fundamental physiochemical mechanism for organizing macromolecules within both the nucleus and the cytoplasm. At the molecular level, weak, frequently transient interactions between molecules with multivalent domains or intrinsically disordered regions, IDRs, drive both phase and separation and condensation. Approximately 30% of the human proteome consists of either intrinsically considered disordered proteins, IDPs or IDRs, occurring within multi-domain proteins resembling beads on a string. In eukaryotic cells, IDPs and IDRs have been recognized as participants in regulated processes central to both homeostasis and disease. In this afternoon's Rutgers Institute of Quantitative Biomedicine crash course, co-sponsored by the Rutgers Cancer Institute of New Jersey and Princeton University, we'll hear from 6 internationally recognized thought leaders in the area. They'll explain how IDPs and IDRs and their condensates contribute to cellular signaling and regulation in eukaryotes, deepening our understanding of the importance of protein disorder and liquid-like assemblies will shed light on cellular organization and may enable therapeutic intervention in protein aggregation diseases by targeting membrane-less intracellular compartments. We look forward to a fantastic meeting this afternoon. I'll now pass the program back to Dr. Burley.
Thank you very much, Ned. So our leadoff speaker is our other co-organizer, Professor Clifford Brangwyn. Cliff received his PhD in applied physics from Harvard University before committing himself to research at the interface between physics and biology. The title of his presentation today is Understanding Membraneless Organelles and Condensates Within Living Cells. Thank you, Cliff. The floor is yours.
Right. Thank you so much. Let's get this teed up. And all right, everybody can see that and hear me fine?
You're good to go, Cliff.
All right. Okay. Well, I want to first thank Ned and Stephen, who really did the heavy lifting on the organizational side of this. I was kind of a consultant, but I'm really glad that this program has been put together. Really is a fantastic the present company fantastic lineup of speakers, and very happy to be a part of this and share a little bit about some of the work that I'm doing. And more generally, I'll just add that it's wonderful to have further interactions. Here in New Jersey already there's tremendous synergies and collaboration between Rutgers and Robert Wood Johnson Medical School and Princeton University, and I'm really grateful for the opportunity to further deepen those connections. I want to tell you about some of the work we're doing, and I think a better title for the talk that I'm going to give today is intracellular phase separation. This is not your parents' lava lamp. Sometimes the analogy is made that this is like a lava lamp in cells, and certainly that analogy has some merit for getting started to think about this. But I want to give you a sense for what are the foundational concepts in this field, but also want to really get across in my talk that this is there's a rich complexity that we're uncovering as we go deeper into this field, and I'll share some of the insights from my lab and the field on that. So as was mentioned in the introductory comments from Ned and Stephen, one of the things that's most exciting about intrinsically disordered proteins is in their role in structuring the contents of living cells, in particular through their ability to drive the formation of a certain type of organelle. Most of the time when one hears the word organelle, we're thinking about membrane-bound, vesicle-like structures, things like the nucleus or the endoplasmic reticulum or mitochondria. That's really just the tip of the iceberg in terms of the way that cells compartmentalize biomolecules. There's a whole class of literally dozens of different types of organelles that do not have membranes, and these are referred to as membraneless organelles or biomolecular or intracellular condensates. These structures are really fascinating because even though there is no enclosing membrane, there's no kind of essentially a sort of enclosing wall that keeps the biomolecules inside, they still are able to assemble and remain as coherent objects in the cell. Many of them are in the cytoplasm. This is we got started in this. I'll show just one brief little slide or panel on these P granules, which are found in the C. elegans embryo. Many others in the cytoplasm, processing bodies, various signaling assemblies, stress granules, and many in the nucleus, which is really the primary focus of our research and what I'm going to tell you about today. They're generally very rich in both proteins and nucleic acids, often RNA and sometimes DNA. And the way in which they assemble is just being uncovered in the rapid developments of this field. So I think probably most people appreciate that phase separation is now, over the last 10 to 15 years, it's being reported everywhere. And I'm happy, I was fortunate to play a role in identifying this process in these structures called P granules, which assemble in the posterior of the embryo, and we showed behave like liquids and have all the signatures of classic phase transition, what's often referred to as liquid-liquid phase separation. But these kinds of phase transitions are now been identified and reported in many different contexts of the cell, in receptor clustering, for example, in these T cell clusters that help catalyze actin polymerization or cytoplasmic stress granules. My own lab has focused a lot on the nucleolus and condensates that are involved in transcription and gene regulation in the nucleus. And that's become quite I'd say quite a central theme of the field, what's happening in the nucleus in terms of heterochromatin organization and activation of genes in Pol2 transcriptional clusters, enhancer promoter interactions, and so forth. So as I mentioned, and really is a central theme of this crash course, is the important role of intrinsically disordered proteins. So we're all familiar with the idea that proteins are linear chains of amino acids, but sometimes even biologists forget that they are polymers because we're used to thinking them as folded entities whose shape and surface features dictates their complementary interactions with binding partners. Many proteins, though, are not folded into single compact shapes. They're actually conformationally heterogeneous, and this is a simulation from one of the speakers today, Rohit Papu. I'm sure he's going to say a lot more about this type of thing. And Alex Holhouse, both of them are at Washington University in St. Louis. These types of disordered, conformationally heterogeneous proteins are important for driving phase separation. In many contexts, if they're purified and one examines them at moderate, physiological concentrations, what you see is that they readily phase separate into liquid-like condensates, things that are akin to what we see in living cells. And these are some examples from my lab, but many researchers in the field are examining the way in which IDPs or IDRs, the regions of proteins, are driving phase separation and how sequence patterning dictates that. We're going to hear about that from several speakers today. One of the things I want to mention, though, in the spirit of the title, this is more complex than we sometimes appreciate. Many proteins that are driving phase separation in cells that are playing key, you could call it scaffolding roles for forming condensates, they contain not only disordered regions, but also oligomerization domains, so shown here in green on the N-terminus, and also substrate binding domains, for example, ubiquitin binding or chromatin binding or RNA binding. I want to emphasize the role of oligomerization, and the idea there is sometimes it's dimerization. Proteins like nucleophosmin that we study a lot in the nucleus has a pentamerization domain. The reason why this kind of oligomerization is important for phase separation is that phase separation one thinks about as a competition between entropic driving forces, which generally want things to be well mixed, so not phase separation, so rather a homogeneous concentration across the sample. But if there are favorable interactions between molecules, that can tend to favor phase separation. So there's a competition between these. Oligomerization essentially cuts down on the relative importance of the entropic contribution. And you can see that in this 1/n term, where n is like the degree of oligomerization or degree of polymerization. This is a classic theoretical framework that people use as a launching point for understanding phase separation, a kind of mean field approach, which is useful but also has limitations. So one of the things that my lab has spent a lot of time on over the last 5 years or so is developing technologies for probing and controlling phase separation in living cells. And the way we do this is by taking advantage of the intrinsically disordered proteins, intrinsically disordered regions of proteins, and we hook them up to oligomerization domains that are light activatable, so optogenetic, light-controlled oligomerization. In this way, we're essentially replacing the endogenous oligomerization, which is regulated through post-translational modification and various biological activity. We're replacing those domains, those oligomerization domains, with light activatable ones. And this has been really powerful for us in the ability to control phase transitions in cells and ask about sequence determinants of the disordered protein drive for phase separation and the impact on function. Here I'm showing you a field of cells. These are U2OS cancer cells that are expressing disordered protein, the disordered protein FUS, in particular the N-terminal region of FUS. And we put it together with this self-assembling core that promotes light-dependent oligomerization around the core. And the bottom line is that when we shine light on these cells, what you see is that the disordered protein readily phase separates in cells. And this has been very useful for quantitatively interrogating the determinants of phase separation and actually mapping phase diagrams in living cells. You can control it in space and time, and ask all about how favorable the environment is in different regions of the cell, and I'll tell you a little bit about some of our work there. That was kind of the background that I wanted, and you're going to hear a lot more about disordered proteins and the basics of phase separation throughout the afternoon session. But I want to tell you a little bit about one of the most interesting and I think functionally important complications, you might call it, to the simplest phase separation picture that is in the review articles and is actually interestingly now in the 7th edition of Molecular Biology, the Cell textbook. So the complication comes from the fact that the cellular environment is incredibly complicated. It is not in thermodynamic equilibrium, so that's one thing that's interesting to keep in mind. It's also incredibly heterogeneous. There's different regions of different mechanical properties and different compositions. And that's particularly interesting in the context of the nucleus, where you have all of these reports of phase separation occurring in the nucleus of cells and having functional regulatory importance and consequence. But we really have not understood how to think about phase-separated condensates, these protein and RNA-rich condensates, and how they're interacting with and impacting the three-dimensional organization of the genome. So this is a cartoon of the nucleus of cell with these different chromatin domains and the organization of the genome. And I've just drawn some spherical droplets here, but we really don't know how to think about this. And I'm showing you, maybe it's something like a spider web with dewdrops on a spider web. That's basically not so far from our, our understanding pictures like that. So we've been trying to push toward developing really quantitative understanding of that relationship. How does phase separation work in, on, and around the genome? Now, some hints of the role of the complex heterogeneous environment in cells in general, and certainly in the nucleus, came from some of the earliest work we did with my first graduate student, Marina Ferrick, who's now a postdoc at the NIH, about to embark on her independent faculty career, where we were studying this frog oocyte system. And the frog oocyte is a really fascinating system because it is a single large cell. It grows to be about 1 millimeter in size, and the nucleus of that single cell is correspondingly large, half a millimeter in diameter or so. And inside that nucleus, there are actually hundreds or up to thousands of nucleoli. They're amplified extrachromosomally, and they're suspended throughout the nucleus. Now, this is a special type of nucleus in that what we were able to show definitively, it was already quite strongly suggested in the literature, but there was lingering controversies, that this actin network is polymerized, and it's a scaffold that stabilizes the nucleoli as a kind of emulsion from doing what your salad dressing does, which is all collect into a single large phase. And in fact, if we disrupt the actin network using the classic perturbations or genetic perturbations, then these nucleoli all basically collapse. They, in this case, kind of amazingly undergo gravitational sedimentation, and you end up with a single large nucleolus, which I think might be the world's record nucleolus. And so we could, we use this system and could understand how the actin network gravitationally stabilizes an emulsion of these nucleoli. So in more typical somatic cells, in the nuclei of somatic cells, rather than actin as this stiff elastic network, what is more the case is that there's a chromatin network, the DNA and associated proteins, that form a viscoelastic material, a viscoelastic gel. And in some work where we've taken advantage of the optogenetic systems to use biomimetic synthetic condensates, these IDR-rich condensates, and we've tracked the motion of them inside the nucleus, we're able to probe these material properties and show that this material, this viscoelastic chromatin network is strongly suppressing the motion and suppressing the coarsening dynamics of condensates inside the nucleus. So there's a rich interplay that's not generally appreciated in the in vitro systems where it's a highly simplified system. The other aspect of this that is something that's been a real focus for us for a while now is the impact of the mechanics, not only on the coarsening dynamics and the ability of these condensates to move around and bump into one another and coalesce, but the mechanical properties of the surrounding network and its potential impact on phase separation itself. So whether or not droplets form. This work began with some inspiration from the group of Eric Dufresne at ETH Zurich, who had been himself, he was inspired by some of the biological systems, but he had developed this biomimetic system based on silicone oil in a gel, and showed that that elastic matrix can strongly suppress and control the sizes of synthetic droplets in a totally non-biological context. We started thinking about that and wondering if something like that would happen in cells. In other words, if a droplet has to grow against a stiff elastic matrix, perhaps the energetic cost of deforming that matrix would suppress and negatively impact the energetics of the phase separation process itself. And so this is something that we've been pursuing over the last several years in several different contexts, beginning with work from our former postdoc Youngdae Shin with a grad student, E.J. Chang, and continued in a quite elegant way from a current student, Yoonji Kim, and a postdoctoral fellow, Amy Strom. So this is an example I'm going to show you of the nucleus of a cell where we have H2B, which is our label that basically is telling us where the chromatin is, where this viscoelastic genomic network resides. And what we're going to do is use this optogenetic system, this biomimetic optogenetic system, to induce droplets to form by shining light globally across the entire cell. And when that happens, you'll see what happens. We form droplets like in the movies I showed you before, but if you look at the chromatin signal on the right, what you notice is that the droplets, as they form, they tend to exclude chromatin. So they're actually forming and they're pushing out the surrounding chromatin network. And you can see that in these still images. What you also might notice from these movies is that in the regions where chromatin is most dense, so in other words, where the chromatin is compacted and generally in this heterochromatin organization, which is full of genetic silence genes, the condensates that we try to form generally don't form there. So we're just activating the system globally, but they tend to not form in that dense region. And that's consistent with this idea that there may be mechanical suppression, so the condensates can't grow to push out chromatin. And so there's this idea of an energetic contribution to phase separation. We can see actually a pretty dramatic example of that in these earlier experiments we did in the 2018 paper, where if you take cells that have these very dense heterochromatin centers, these chromocenters, and we shine the light specifically on that chromocenter and try to induce phase separation in that dense network. And here's what happens. So what you see in this case is that phase separation just will not occur in that mechanically dense region. It instead forms these flower petal-like arrangements around the outside. And that could actually be pretty much exactly recapitulated in coarse-grained simulations that my colleague Miko Hataya and a former postdoc of his, Joel Berry, did. So this idea that nucleation and growth is suppressed in a mechanically stiff environment. That seems, one critique one could make is, okay, this is a biomimetic system with IDRs. Maybe this is very different than what's happening natively in the cell. So it turns out, and we're, I've only shown you a couple different IDRs. Some of these were FUS and some of them were BRD4, the disordered region of BRD4, about which I'll say more. But it turns out that generally it's the case that if you overexpress tagged phase separation prone proteins, in particular proteins that have significant disordered regions, what you find is that they form these condensates, and the condensates tend to exclude chromatin, which you can see in the H2B signal. We get this sort of black hole where the condensate forms. Not only that, but, and because of course that's an artificial system too, we're overexpressing proteins. But if you do immunofluorescence and ask the various condensates that occur naturally inside the nucleus of cells, what is their relationship to chromatin? Generally, also the case that they tend to exclude chromatin. That the region inside those condensates, certainly the nucleus and these other examples, exclude chromatin. So there's a very low signal from H2B, and we see that validated with if we do something like Hooks or DAPI. So this idea that chromatin is excluded from condensates, and that's the disordered region is sort of sufficient for doing that, is kind of interesting. Of course, we know that many proteins that have significant regions of disorder have specific chromatin targeting domains that bring the protein to a specific genomic element. For example, various epigenetic marks, acetylated lysines in the case of the bromodomains on BRD4, or other kinds of targeting domains. So what we were wondering is, what happens if we do that? What happens if we specifically target IDR-containing proteins or native proteins that have chromatin binding? Does it, form a surface coating of chromatin, or does it pull chromatin in a sort of compaction? How do we understand that, and how do we think about that as well in the context of this sort of mechanical picture? So this is some unpublished work that is being driven primarily by Amy Strom, Urena Eftons, Yoonji Kim, and a former postdoc who's moved on, Chanel Jumper. And this is now this protein BRD4, which is a well-known protein that's involved in activated transcription in cells and Pol2-mediated transcription. And if you look at endogenous, so this is an immunofluorescence of BRD4 in the nucleus of a human cell, what you see is these puncta throughout the nucleus that form. And we were wondering, why do you get these very small puncta? Why do they not coalesce into the larger droplets that we see? For example, if we just take the disordered region of BRD4, in other words, lop off these bromodomains that bind to acetylated histones. And I'll just show you. So the picture, of course, is that there's suppression. Somehow you've got something like nanodroplets that form at these targeted sites, but they're not coarsening because they can't meet each other because of mechanical constraints. This is this picture on the right is just showing you we basically get the same structure that forms if you overexpress BRD4, M-cherry tag BRD4. So it sort of validates the ability to learn something from that because it looks just like the native immunofluorescence. Yeah, and so I've got an animation here showing this disordered region pops up, this disordered region, which is sometimes referred to as delta N because you're lopping off these bromodomains. So a cool trick is there's a drug called JQ1, which specifically targets the bromodomains and the interaction of the bromodomains with these acetylated lysines on histones. And if you introduce this drug JQ1, what you see is that you basically get pretty homogeneous signals. So you no longer see punctate BRD4 signal. You will see some larger ones, and that's kind of interesting. That pretty much looks exactly like the BRD4 delta N construct. So this is the one that doesn't have the bromodomain. So it makes sense that it looks something like the full length in the presence of the bromodomain disrupting drug. So we had this idea, maybe what's happening is, yeah, you've got these nanoscale droplets that aren't coarsening, but then they are dissolved away once you disrupt that interaction. What's interesting though is that there's a concentration dependence to this effect. So if we overexpress BRD4, this M-cherry BRD4 construct, in the presence of JQ1, if you go to higher concentrations of the full-length BRD4, you start to get large-scale droplets. Again, consistent with the idea that natively what's happening is you're suppressing coarsening due to the chromatin targeting. And that's basically exactly what you see, again, in the BRD4 delta N construct, the construct that does not have the bromodomains that are targeting chromatin. And so you can quantify all that. You basically get in the presence of JQ1, you get a significant decrease in the number of condensates that are counted in the nucleus, and that's exactly consistent with the delta N construct. So we think this coarsening is kind of a central part of it. Another interesting aspect of this we started thinking about, and this is builds from some earlier work that was just published really last year from a former postdoc who's just moved on to Kyoto, faculty position in Kyoto. So what Shun did was map the phase diagram using this correlate optogenetic system, as we've done several times in the past. This core to IDR ratio is essentially the degree of oligomerization, which, because it's an entropic effect, we think about this essentially as a temperature-like axis. This axis is the concentration of the core. And you can see that what separates the cells that do not phase separate, these triangles, from the ones that do, the circles or the squares, is this line, which we refer to as the binodal boundary. And this is a sort of a finer point that's referred to as the spinodal, which separates nucleation and growth from spinodal decomposition. But what Shun showed is that as you go deeper and deeper into the two-phase region, in other words, as the system becomes more and more supersaturated, the nucleation rate goes up and up and up. Moreover, what's interesting is Shun showed that we could specifically engineer heterogeneous nucleation sites. In other words, sites where the surface interactions are energetically favored, such that when we induce phase separation by turning on the light, we only get condensates that are forming at these favored engineered sites. So in this case, these are telomeres that have been engineered to express, to have FUS attached to them such that when we form these de novo FUS droplets, they always nucleate at the telomere. But there's a strong concentration dependence. If you highly supersaturate the system, you basically overcome that energetic favorability to undergo heterogeneous nucleation. This whole paradigm, we started wondering if it would be useful for the BRD4 system. And in particular, there was a prediction that in the presence of this drug, JQ1, that specifically targets BRD4 to acetylated lysines, these epigenetic marks, what we might see is a difference in the ability of those marks, those acetylated lysines, to promote heterogeneous nucleation of BRD4 on particular genomic elements. And in particular, the prediction, this is just schematic data saying what we expect is that without JQ1, you've got these heterogeneous sites and you expect rapid nucleation. The delay time for nucleation and the slope, should be much more favorable for nucleation than in the presence of JQ1, where you're basically waiting for more like non-specific homogeneous nucleation. I'll put that in quotes because there's almost no such thing as homogeneous nucleation, but certainly not specifically targeted to these acetylated lysines. And it turns out that's basically exactly what we see. So in the presence of JQ1, when we do this optogenetic activation experiment, we only get droplets that are forming much later in time. So there's a significant delay in the nucleation, again, because that chromatin binding domain is not able to target. So that's kind of interesting and suggests that there's this close interplay between phase separation and specificity of binding and specificity of nucleation and growth of these condensates. So it's there's certainly a programmed logic, or as Rohit likes to say, grammar associated with the interactions and their favorability for phase separation, where it occurs, when it occurs, and so forth. The other part that I want to speak about just in the last couple minutes is this idea that condensates can generate force in living cells. Now, I have made all caps the real forces because I have spoken about these ideas before, and people sometimes say, well, you don't mean like real forces, it's some kind of analogy. And I'm saying these are real forces. Like they can do work, and we could power a little micromotor or something. So the idea here is that capillarity, the energetics associated with the surfaces of liquids and their interaction with other surfaces can actually generate real force, measurable force in units of piconewtons or higher. So this is something that we're putting forward as an idea because capillary forces are actually absolutely ubiquitous in nature whenever you have liquids around. And turns out now we understand there's a whole bunch of liquid interfaces in living cells. So this is something that we're really excited about, this idea that you can start to think about condensates as little molecular motors that can actually generate force in cells. We had some hints of this in our earlier work, and Yunji and Amy have really turned this into a paradigm for engineering force generation within living cells to actually probe mechanics and function in really clever ways. These are telomeres, which is, again, one of our favorite sites for genomic manipulation because it's easy to target things to them. And so what's happening here is that we are writing droplets, we're writing condensates onto the telomere, and I'm going to show you an example of that. We basically put these FUS condensates on the telomere, and then we try to write a droplet by shining the light between these two separate droplets. And this is what you're going to see in the movie. Here are two telomeres, and there's condensates forming. Then we bridge them with light. And then what we do is slowly turn off the light. And as we do that, the condensates, by some miracle, remain attached to the telomere surface, but shrink and actually pull together those different, those distal genomic elements. So that's a real force, and it's pulling against the viscoelastic matrix of chromatin, and it's something we're really fascinated by. And it turns out you can learn a lot about the material properties of the nucleus and different aspects of what's happening functionally when we can on demand pull different parts of the genome together. So I'll just wrap up with a couple comments here. First of all, there's a rich complexity of condensate genome interactions, all kinds of interesting things happening. It's not as simple as phase separation in a buffer. Condensates are excluding the genomic elements, but they can also target it, pull it together. They can compact the genome in heterochromatin. I didn't even talk about some other work we did with Urena Eftons published last year. But there's a rich complexity that really this is just the very beginning of studying this, and the implications for mechanobiology, in particular genome compartmentalization and its role in mechanics of cells and metastasis, where cells have to crawl through neighboring tissues in order to do the dirty work of malignant transformation and spreading. That's something that I think is a really fascinating intersection with phase separation and genomic organization. So this is definitely not your parents' lava lamp. That's the take-home message. And I think I thanked most of the key folks that I've worked with on all of this, done all the hard work in the lab over the years. Here's a list of some more folks and a picture from not that long ago. And I'm happy to take any questions. Thank you very much for your attention.
Thank you very much, Cliff. That was a tour de force. I'm monitoring the chat for any questions that those of you would like to have me direct them to Cliff. And the first of these comes from Richard Ludischer. Do these condensates undergo Ostwald ripening?
Yeah, this is a question that we talk about probably 2 or 3 times a week, probably. What's interesting is I'm going to say something maybe a little bit bold here, and somebody will probably tell me I'm wrong, but I'm unaware of any unambiguous example of Ostwald ripening in condensates in living cells. There's examples where, for example, even in the P granule thing from our 2009 paper, where there's gradients of stability that are set up in the system, and you might stretch the definitions and say that's a kind of Ostwald ripening. But the reality is Ostwald ripening rarely is observed, if ever. And part of the explanation for that, I think, is that the surface tensions are very small. Because these are macromolecular liquids, the surface tensions are small, and micronewtons per meter and so forth. Usually coarsening occurs through Brownian motion coalescence, where they actually bump into each other and grow that way.
Thank you, Cliff. James Gregory asks, does inducing droplets in the nucleus change the organization of the genome in 3D?
Yeah, that's something that, I showed you in the microscopy images that, there's clear structural changes, right? There's clear holes that are being formed and so forth. So you might ask, I think this is probably what you're getting at, what happens to TADs and insulators and the chromosome territories and all of that. That's something that there's some nascent work asking those questions, taking high-C and approaches like that. We actually have quite a bit of work growing on using the Sprite technology, which was developed in the Gutman lab by Sophie Kinados, who's now a postdoc in my lab. So that's something that very little has been clearly elucidated in that context, but I think it's an exciting area, and we're certainly thinking a lot about that as well.
Thank you, Cliff. One of my Rutgers colleagues, Monica Roth, asks, does the ET domain associate with other chromatin binding proteins, and can those be seen outside droplet but on the chromatin?
I don't know the answer to that question. I don't think we have looked carefully at that. But yeah, I'd have to think about more about that. I would encourage you to, if you have specific suggestion on an experiment we should do, please send me an email, and that would be greatly appreciated. Thank you.
Thank you, and I regret that we don't have time to Cliff, for you to tackle additional questions that are in the chat, and I would ask those of you who do have further questions for Cliff that we weren't able to get to, just simply email him, and I'm sure he'll be happy to oblige. So with that, Cliff, I'll ask you to stop sharing the screen. Thank you again. That was terrific. Our next speaker is Dr. Rohit Papu, the Jean K. Beer Distinguished Professor in the Department of Biomedical Engineering in the McKelvey School of Engineering at Washington University, St. Louis, Biomedical Engineering. After undergraduate training at Bangalore University, Rohit received his PhD in biological physics at Tufts University. His title today is Theory of Associative Polymers and Implications for Biomolecular Condensates. Rohit, the floor is yours.
Thank you, Stephen. Can you hear me?
We can hear you and see you. You're good to go.
Perfect. Thank you very much. So it's a real pleasure to be here. Thanks to the organizers for putting together this event. It's always a tough act to follow Cliff, but actually he teed this up very nicely for me. I think the gist of his talk can be summarized by two important comments. There's hidden complexity, and the concept of viscoelasticity is a very important one. What I'm going to try to do is extend those two concepts down to the simplest of systems to point out that there are hidden complexities even in the simplest of systems. So it's not just in the cellular milieu that the hidden complexities that Cliff referred to are manifest. So a theme that Cliff may have glided by mentioning is this concept of multivalence, the idea that macromolecules that drive phase separation are in fact multivalent in nature. And I will start with the motivation that multivalent macromolecules, the ones that we care about, proteins and RNA in particular, are biological instantiations of what in the polymer literature one refers to as associative polymers. So what are these associative polymers? These are basically macromolecules with attractive groups, which perhaps doesn't say much. So let's think a bit about this a little more formally. So essentially one can think about associative polymers as containing domains or motifs or even individual residues that act as stickers that can participate in reversible physical crosslinks. The simplest such interactions would be reversible hydrogen bonds, ionic interactions, cation pi, pi pi, and so on. And one can realize a gamut of such reversible interactions either just by the linear architecture of the chain or through the types of topologies that are enabled through folding, for example. And the spacers are going to sort of participate here as facilitators, not as passive players, but as facilitators or inhibitors of these specific interactions. So basically, the first point to get across is that associative polymers don't come in just one flavor, they come in a variety of flavors, and they drive the formation of strong or weak networks of physical crosslinks. And this concept of a network becomes super important, especially as we pivot toward ideas like viscoelasticity. So here are a few instantiations of sticker and spacer architectures for proteins. For example, there is no hard and fast rule that says that the drivers of phase separation have to be intrinsically disordered proteins. One can very well see these even with things like glycolytic enzymes, where you have essentially fully folded proteins that can oligomerize, a point that Cliff touched upon, and the hotspots on these surfaces can in fact drive specific interactions. Now in this context, the blue surface of the folded domain essentially acts as the spacer. One can, of course, go to the opposite end of the spectrum and look at systems such as the preon-like domain that Cliff touched upon as far as FUS was concerned, where now you have a linear distribution of stickers, basically uniformly distributed along the linear chain. There are the systems that Mike Rosen popularized in terms of polyvalent systems whereby folded domains are connected by disordered linkers. And in fact, here it's the synergy between the ordered and the disordered regions that will sort of contribute to the driving forces for phase separation. In this particular case, it is the specific interactions between the proline-rich modules and the SH3 domain that provide bulk of the driving force for phase separation. And of course, the classical examples of heterotypically driven phase separation would be a concept called complex coacervation, where one has got linear chains of oppositely charged macromolecules coming together and making what are called coacervates. All of these fit in beautifully with the sticker and spacer architecture concept. So what I'm going to do is, before I jump into data, basically tell you about the underlying important conceptual clarity and complexity that sort of drives phase transitions of systems with sticker and spacer architectures. If all you focus on is the multivalence of specific interactions, then basically what we have in play is the possibility that our molecules can make dimers or trimers or tetramers or higher order oligomers. It turns out that there will be some threshold concentration above which essentially the molecules make these reversible networks or reversible crosslinks that can generate a system spanning network, which in the old days used to be called a gel, before the unfortunate event happened of the condensatology essentially corrupting the term to mean a bad thing. So we'll basically refer to this as a system spanning percolated network, meaning the network has taken over the entire system volume. But it turns out that, of course, there's also an interplay between the polymer-polymer and polymer-solvent interactions. And so the incompatibility of polymer-solvent interactions, which we capture using the free energy of sort of essentially phase separation or mixing, the Flory-Huggins term. So here is this chi term, which basically measures the extent of the incompatibility of the polymer and the solvent. If you layer that atop the contributions of specific interactions, what you can see is a coupled transition, whereby now the system separates into two coexisting phases, a dilute phase, which has some sort of bulk concentration now we'll refer to as CSAT, and a dense phase, but the dense phase is not some random agglomeration of the molecules of interest. In fact, for associative polymers, what they are going to be is a networked collection of these molecules. That is typically because this type of an inequality is satisfied, whereby the dilute phase coexisting concentration CSAT is at one end, the dense phase coexisting concentration is at the other end, and essentially inside of the condensate or inside of the dense phase, we cross this percolation threshold. So in fact, the optimal description of condensates would not be of liquids or simple liquids. The most general description would be of physically cross-linked, what are called non-affine, which means that they have sort of anisotropic deformations to mechanical forces, network fluids that are viscoelastic in nature, or what Tom Vilges in the old days used to refer to as a microgel. Okay, so this brings up a really important point, which is that there are going to be a spectrum of properties that are going to show either monotonic or non-monotonic variations between sort of the viscous and elastic limits. One can think about a purely viscous fluid. It's going to be governed primarily by the sticker to spacer ratio. The purely elastic limit would essentially be a polysticker in some way. There are moduli that essentially tell us how much energy you can store and how much energy is dissipated. That ratio of the storage to the loss modulus is also tied to the sticker to spacer ratio. And likewise, the interfacial tension, which Cliff alluded to as being very low for a given macromolecule size, basically will start to sort of plummet down as we head toward more sort of elastic network pictures. So this leads us then to an important point, which is that we're not describing canonical phase separation or LLPS. We're really describing phase separation coupled to percolation, which requires the consideration of a diverse set of energy scales. So let's step back a little and think about sort of phase separation 101. All of the magic sauce is in this polymer solvent compatibility or incompatibility. which is defined by this chi parameter. It's basically quantifying the balance of the protein solvent or polymer solvent, polymer-polymer, and solvent-solvent interactions vis-a-vis KT. An ideal mixture is one where chi is 0. In a poor solvent, chi is positive, and that's when you're likely to see phase separation. Now, it turns out that when we layer upon this generic chi, which sort of contributes to the Florey-Huggins expression for the free energy, a lot of specific interactions, which we'll refer to as the free energy of associations, the ability to make stoichiometric and non-stoichiometric assemblies, then we end up essentially renormalizing our chi such that our chi now becomes the original chi. So essentially, let's pretend that we sort of took out all the stickers, to which we now make some changes whereby we say that there will be concentration dependent contributions. That's what phi is. Phi is the volume fraction of polymer in a simple polymer solvent system that will sort of modulate through the contribution of specific interactions, the expectations for the solution as we change concentration. So this turns out to actually have some very directly testable consequences. So I should point out that these are comments and things that we've been making a lot over the past few years, but what we were missing were some direct tests of the distinction between an LLPS picture versus a phase separation coupled to percolation picture. So let's start with something really, really simple. There is this threshold concentration we refer to as the saturation concentration. It came up in Cliff's talk. The key point in a purely phase separation only picture, where we have only one energy scale, is that the subsaturated or the undersaturated phase, and Cliff pointed out that this is seldom true, the undersaturated phase should be made up of primarily dispersed monomers and nothing else, right? And so in fact, this turns out to be borne out in simulations. To this point, we have sort of developed a Lattice Engine, which will feature again during the talk. We call this LUSSI. The primary developers were a former postdoc, Zhongwo Che, and a current graduate student, Furkan Dar. So what Furkan did was he said, well, let's take a homopolymer. All of the interactions are exactly the same. That means the only interaction prevailing here is chi, right? What he's doing here is quantifying the probability of realizing a cluster of some particular size. The dashed line represents essentially the supersaturated solution. You don't see anything corresponding to the subsaturated solution here, because essentially in this simulation, the system is completely dominated and overwhelmed by nothing but dispersed monomers below CSAT. So notice no blue colors, no green colors, no yellow colors. Let's just add a little color here. What we'll do is we'll basically say that there will be some stickers that are going to be capable of specific anisotropic interactions, right? They can make reversible crosslinks. Now what one starts to see is that as in the undersaturated or subsaturated regime, there is a finite probability of forming clusters of different size. Importantly, as we increase the concentration, so as we are starting to approach CSAT, the probability of realizing a cluster of a particular size actually increases, rather like an isodesmic process, right? This is exactly what you see in a sol-gel transition in the sol regime. What this means that in the subsaturated regime, you should see a series of finite sized clusters. They're not going to be system spanning. They are going to have finite translational entropy. And the picture that emerges then is that CSAT is really the threshold above which these clusters network to form a viscoelastic condensate. So there's a prediction that comes out of this, which is that the cluster size distribution in the absence of a coupling to percolation should be a bounded distribution for something like pure LLPS, shouldn't really change as a function of concentration. But as we add associative interactions, so there's this separation between the solubility determining and the specificity determining interactions, as the concentration increases toward the saturation concentration threshold, The cluster size distribution has this heavy-tailed character, meaning that the overwhelming number of species will be small merged, but in the tail of the distribution, you'll start to pick up mesoscale species, right? Can we observe this in through direct measurements is the question. For this, we'll pivot to these RNA binding proteins or FET family proteins. And unlike what Cliff was talking about here, we're going to talk about the full length proteins. So this includes the arginine-rich RNA binding domains that also have folded RRMs and the intrinsically disordered prion-like domains. I'll focus exclusively on FUS, but please note that what I am sharing with you here today transfers very nicely onto all of these other proteins. So one of the things we had discovered a few years ago in collaboration with Tony Hyman's lab is the network of arginine tyrosine interactions basically are the primary stickers that help drive phase separation of FUS and FET proteins. So here is a simple sort of experiment in buffer. So essentially the idea is that as you crank up the concentration of FUS, there appears to exist a threshold concentration known as the saturation concentration, above which one sees these droplets forming. One can be extremely rigorous about nailing down this CSAT value for a given set of solution conditions. Now we'll pivot to asking what happens below CSAT in the subsaturated solutions. This is the work of an extremely committed and talented postdoc, Joy Carr, who works in Tony's lab and who's forged a beautiful collaboration over the past 2.5 years with Furkan Dar, PhD student in my lab. Along the way, we needed more techniques to sort of be brought to bear, and we were really fortunate to collaborate with Klaus Seidel and Thomas Knowles, and it's always good to have Simon Alberti's critical eye on this story. So here I'll start with Klaus's data. The threshold concentration for FUS-SNAP, and by the way, all these experiments are done with tags and without tags. I'm just showing you the tagged versions here. In this case, Nile red is the fluorophore that's basically running around in solution, and its anisotropy in the presence of polarized light is being read out as a measure of what is happening as we titrate the concentration. Notice that the anisotropy is basically increasing monotonically as we titrate the concentration of FUS-SNAP by basically 3 orders of magnitude. But remember, I told you that if such clusters were present, the underlying distribution should be heavy-tailed, and that distribution should have a shape that is very much concentration dependent. So Klaus had developed this method called fluorescence intensity distribution analysis, which you can essentially use to effectively sort of bin the data. And from those data, he can essentially show that as the concentration increases, at very low concentrations, the subsaturated solution is really dominated by small species. But as the concentration increases, we start to take on these heavy tails, meaning that we're sort of graduating the distribution of clusters to take on larger species. Which means that there should be DLS detectable species as well. Because what is DLS good for? Let's just imagine that we have a heavy tail distribution. It should see the most abundant species, namely the monomers, but it should be very sensitive to the presence of a tail because of the R to the 6th weighting. And so it should also see mesoscale species. So not surprisingly, as we increase the concentration, one actually starts to see the appearance of these mesoscale species. And these are not small. These are not dimers, trimers, tetramers. We're talking about hydrodynamic diameters that essentially allow the encompassing of hundreds of molecules, but they are not the only species, right? I mean, this is a true mixture. The important point is that these clusters are reversible, so one can essentially dilute the DLS intensity grams change. One can essentially reconcentrate and then reacquire the signal. Now comes the really important point, which is what is the relative abundance of the larger species, so hundreds of nanometers in diameter versus the smaller species. For this, essentially one turns to something like nanoparticle tracking, where you take the advantage of scattering and you couple it to the advantage of dark field microscopy that allows you to track the Brownian motion of scatterers, an experiment that sort of looks like this. So here in deeply subsaturated solutions, basically you see these clusters, and notice that the abundance increases as we increase the concentration by a factor of 2. These are all below CSAT. You see these clusters sort of running around. One can quantify the abundance at a series of different concentrations. Notice that in accord with the theory, you essentially see that these distribution sizes shift to the right, meaning they take on higher sizes. From these data, we can calculate the relative abundance of mesoscale clusters, right? And that makes, so these large species that are scattering detectable make up about less than 1% to 1% of the solution. You might sneeze at that, but from a nucleation standpoint, it's completely right. And so, but also there's another important point, which is what then is CSAT, right? So in a phase separation coupled to percolation, the idea is that the translational entropy of the largest clusters will start to become really small, which means that you will start to see a coarsening transition. Can we see that? So here are DLS data plotted in terms of the autocorrelation functions. You essentially see below the CSAT, they reach a steady state profile. But above CSAT, you can actually start to see the appearance of a slow mode, whereby at longer time scales, you start to see things actually growing. Now, this can come about due to two possibilities. One is that there's a coalescence of clusters that are actually growing, or you could sort of imagine that we're really probing the motions of polymers in sort of dense phases. But the way you sort of settle in favor of condensation by coarsening is really in terms of light microscopy, and you actually start to see that there are small things sort of latching onto big things and so on. So these pre-percolation clusters are present. It basically says that there is a contribution from a hierarchy of interactions that are specific versus solubility determining, and these should be separable using solutes versus mutations, right? I'll show you the solute story. One of the things that you hear of a lot in the literature is the use of hexane diol. And it turns out that actually the addition of hexane diol causes a loss of the slow modes, the disappearance really. Similar data are observable with ATP, which is a hydrotrope. But the key point is that the clusters basically stay immutable or untouched by hexane diol. So what this part of the story says is that pre-percolation clusters will be present. These are present at ridiculously low concentrations, may well be concordant with the endogenous concentration. The important idea then becomes that the cluster size distributions, the structures of these clusters and the reactions of these clusters may well be of functional relevance. And the important point is also that the mutations can disrupt this cluster, this coupling between clustering versus phase separation via coarsening. Now, what I will do is I'll sort of pivot in the last few minutes to one other story, which is also just as exciting, I think, which is this interplay of sticker and spacer interactions in an archetypal intrinsically disordered domain, the pre-on-like low complexity domain. This is derived from work that we recently did with Tanya. Tanya is going to go over this in some detail. Anna and Wade are postdocs in Tanya's lab, Bina a graduate student, MD PhD student in my lab. So here is the archetypal pre-on-like domain that we use, the low complexity domain from the protein HNRNPA1. So notice that the or low complexity domain is shown over here, and there's nothing low in complexity about the system is what I want to guarantee you. So basically what emerges over a few years of work is that in this case, the stickers are primarily the aromatic residues in this case, right? Arginine in this particular case is really an auxiliary sticker. Glycine and serine are the main spacers. And here is an important point. Too often we find ourselves hearing people make statements like intrinsically disordered proteins engage in weak, non-specific, heterogeneous interactions. Well, actually, IDPs are plenty good at making sequence-specific interactions, and really the specificity is governed by the sticker-to-spacer ratio, the patterning of stickers versus spacers, and the types of stickers versus spacers. So what Mina did was he essentially took data in the single chain limit as collected using small angle X-ray scattering, developed a machine learned based model. This is a physics-based model, but the parameters are gained from machine learning. And you see that the parameterization sort of indicates that there's a hierarchy of interactions. So the tyrosine interactions are the strongest, followed by the tyrosine phenylalanine, followed by phenylalanine, and so on. So there's clearly a hierarchy of interactions. They're not all exactly the same. What he can do then is compute binodals, so the underlying phase boundaries in this temperature concentration space. The solid symbols are what he computes. The open symbols are what Tanya's lab has measured, and there's very good agreement. I use this mainly to pivot to answer two questions. These simplest low complexity domains, it turns out actually have a very, very interesting both condensate structure and interfacial properties. This is the work of Mina Farag and Cohen, a postdoc in the lab. So first and foremost, it turns out that, and I'll finish this in a minute and a half, the condensates, sort of topologically speaking, at a given temperature, actually have what we refer to as a small world network structure, meaning that here each chain is a node in the network, and the number of edges that sort of emanate from a particular chain or node represent the degrees. And the key point is that notice that there are only few hubs, right? And there are lots of spokes, but there are only few hubs, meaning that there is this sort of underlying parsimonious or small world structure to the networks, which changes as we go well below the critical temperature, or we approach the critical temperature. As we go well below, you start to see more hubs forming, which can also potentially give rise to this kind of jamming transition, right? Now, the other point to make is that where is this sort of small world network coming from? It turns out that on average, the chain conformations are actually quite different across the phase boundary. Here is A1 LCD. We're plotting data against temperature. The chains are way more, or not way more, are more expanded in the dense phase when compared to the dilute phase. And what this is facilitating is basically the ability of the chain to make networks, intermolecular networks, with other chain molecules inside of the condensate. That is depicted here by the fact that here is a random chain picked out from the middle of the condensate, and each color is supposed to indicate if the chain is, or the crosslinks are being made with different chains or the same chain, and in fact, they're coming from different chain molecules. Now let's sort of end on what then happens at the interface. So what Mina went back and asked was, he can quantify the radial density profile going from the inside of the condensate to the outside. We can fit to Ben Widham's theory of a hyperbolic tangent function for the structure of the interface, and that fit is actually very good. So we can actually now predict the interfacial width quite nicely. And we can follow the conformation of a chain molecule as it goes through the interface. And notice this non-monotonic dependence of conformation. It's expanded, it sort of falls and gets even more expanded as it comes out. That is mainly because it's losing intramolecular crosslinks, thereby setting up essentially a structure that looks like this, right? So what's happening is that you've got a network. As the chains sort of exit the condensate and come out, they point roughly perpendicularly to the interface and then come out and sort of compact on themselves. So what ends up happening is that we create an interface that basically has the property that we have a lot of unsatisfied stickers, potentially priming the interface for reactions and even fibril formation. So the hope is that I've tried to rapidly convey to you, there is both a network structure that underlies the viscoelasticity of even simple condensates. And I'll stop there. I see Stephen has come on. It's gone over just a bit. And thank you all. I think I've thrown shouts out to people as I've gone along. And I'm happy to take whatever questions there are time for.
Thank you very much, Rohit. Terrific talk. We only have time for one question, and I'm going to allocate that to Sukbum Hong, who asks, will pre-percolation clusters form at favorable nucleation sites if the associative polymer possesses some kind of substrate binding domain?
Yes, absolutely.
Okay, thank you. That's a very quick answer. So Rutgers colleague Vic Nanda asks, is there a limit to the number of condensed phases that can coexist?
Yeah, I see the question. Yeah, absolutely. So I think what you're asking is a very, very general question. So essentially what Vikas is asking is if the chemistry will sort of formally impose a limit on the number of phases that can coexist. I haven't thought about it that way, but that's actually a very interesting way to think about the problem. Certainly, yeah, and I had not thought about it that way, because certainly I can come back to you with a more intelligent answer.
Thank you very much, Rohit. And if you don't mind looking at the other questions in the chat and responding, that would be much appreciated. So our next speaker is Dr. Amy Gladfelter, Associate Professor of Biology at the University of North Carolina at Chapel Hill and 2016 Howard Hughes Medical Institute Scholar. Amy received her PhD from Duke University in Durham, North Carolina, which must make basketball season somewhat challenging. Her title today is How RNA Encodes Properties of Condensates. Thank you again, Amy. Floor is yours.
Okay, fantastic.
You're good to
Can
go.
you hear me okay? Can you hear me okay?
Good to go.
Okay, great. Yes, it's just a real honor and pleasure to be here today to talk about the role of RNA in biomolecular condensates. Despite the title of the course, I hope that this talk piques people's interest in thinking deeply about the molecular roles that RNA play in biomolecular condensates. And so specifically, I'll talk about work today we've done using the SARS-CoV-2 virus to uncover the role of RNA sequence, to understand phase separation, and specifically temperature-dependent phase separation of the nucleocapsid protein. But before I get into this data, I want to first just introduce us and bring us into really a common mindset for thinking about RNA and condensates. So we know that many condensates contain RNA. They play roles in the transport of RNAs in large cells, the protection of RNAs under stress, and the positioning of RNAs, for example, during development. And we've just heard two fantastic introductions to really what the nanoscale molecular interactions are that often drive condensate formation for proteins. We've heard about how both intrinsically disordered sequences as well as ordered oligomerization or protein interaction domains can encode stickers that will allow for specific interactions that can ultimately lead to a density transition of molecules and the emergence of condensates that we can detect in the microscope. What I really want us to be thinking about is that, of course, as we've just heard, the strength and the spacing of these stickers is going to be very important for informing the material properties and when and where condensates form. But what I want us to think about is, just like proteins have this ability to control phase behavior through specific molecular interactions, RNA also has critical components that are very specific. And I want us to think about the stickers and spacer model that we've just heard so much about from Rohit in the context of RNA. And that's really what I'm going to be focusing on today. So first, let's just think about the general properties of RNA that can contribute to condensates before we think about specificity. So for example, we know RNA, unlike peptide components, provides a uniform negative charge. RNA can provide multivalency through the system through both specific primary sequence and structural elements, which we're going to be talking a lot about today. But RNA also gives highly variable and also very large sizes compared to proteins of the molecular constituents. And RNA can also take on a wide array of highly dynamic structures that include both structured and unstructured elements. And then finally, RNA can bring distinct mechanical properties with different persistence lengths and essentially different flexibilities compared to the protein components. So there's a myriad of traits that RNA can essentially contribute to condensate physical and specific properties. But what I want to talk about today is what we're learning about what the functional accelerators and spacers are in RNA. And viruses, and in particular the SARS-CoV-2 virus, has been a phenomenal guide in trying to decipher what RNA stickers look like. And that's because in this very, very obviously important virus is a very limited proteome. There's only 24 proteins that have to carry out this entire complex replication cycle. And I think biomolecular condensates and specific assembly factories are becoming well appreciated for a way in which this complex reproductive pathway can be undertaken within the host cytosol. And of course, we have a couple of players here that we think are very important for contributing to viral replication. There's the nucleocapsid protein that I'll be talking a lot about today, which has both disordered structure, it has an oligomerization domain, as well as 2 different RNA binding domains. And genome and actually subsequent subgenomic RNAs that are important in this replication process. And so I'm going to talk, actually, first I want to just show you that in fact, the nucleocapsid protein, based on, as I just showed you, it's disordered, it can bind nucleic acid. Indeed, it can undergo phase separation in cells. N protein, you can see in this cartoon here, is closely associated with the genomic RNA. It's very important for packaging this, the genome of SARS, but it's also probably very additionally important in various steps of viral replication. So N protein on its own, expressed in mammalian cells, forms these large macroscopic, spherical and fusible condensates. And in fact, in SARS-CoV-2 infected cells, when N protein is visualized, you can see it's also present in these macroscopic condensates that are in the vicinity of nuclei associated with the endoplasmic reticulum. So I'm going to talk about 3 different stories that have emerged from studying N protein condensation that's given us great insight into understanding RNA-based stickers. I'll first talk about the role for double-stranded RNAs in setting the temperature of N protein phase separation. So these are RNA structure stickers that play a role in eliminating or breaking apart a dimerization domain in N protein. I'll next talk about a role we think for primary sequence stickers in the RNA that we think may be relevant in giving us insight into how RNA is actually packaged in the viral life cycle. And then finally, in just the last minute, I'll talk about another kind of sticker that we think could be emerging in RNA-RNA interactions and intermolecular interactions. So let's first look a little bit more closely at the nucleocapsid protein. So as I, excuse me, just as I already mentioned, it has an intrinsically disordered region, and it has 2 distinct RNA binding domains, RNA binding domain 1 and RNA binding domain 2. And interestingly, overlapping with RNA binding domain 2 is a dimerization or oligomerization domain. The other key player here is this magnificent RNA genome. The genome is 30 kb. This is a gigantic molecule. We're only looking at a 30th of it here. It's a highly structured RNA element. And some of the first experiments we did on this was to see if we could reconstitute condensation by mixing these two molecules together in vitro. And indeed, we could see that the nucleocapsid protein, when mixed with an RNA containing genomic RNA, we could see readily this nice concentration-dependent demixing. And the N protein on its own showed very little condensation. One of the first experiments we did was actually assess the temperature sensitivity of phase separation. And what we could see is that N protein on its own, while at room temperature or at body temperature, was not able to undergo phase separation. At higher temperatures, we could see it readily formed liquid-like condensates. But interestingly, when we added viral RNA sequences to this, we could actually lower the temperature at which this phase separation occurred. So this is very interesting. One, it shows that N protein shows LCST, or lower critical solution temperature behavior, which is fascinating unto itself, but also that RNA can actually help contribute to the LCST behavior. So how does RNA lower LCST? We approach this by actually making a variety of different mutants in viral RNA and assessed what impact it had on the phase behavior. And one of the most clear observations that we have is that addition or increase of double-stranded RNA sequence could actually lower the LCST. So we could see phase separation at much lower temperatures, even down to 24 degrees. Now, notably, this isn't sensitive to the specific RNA that we added. In fact, it could be any kind of sequence here. It was simply a matter of increasing the amount of double-stranded RNA that's present. We did this for a large number of constructs, and we could see that increasing the double-stranded RNA content could exquisitely tune the temperature required for condensates to form. So this is extremely sensitive to the amount of double-stranded RNA. So how is double-stranded RNA changing the temperature for N protein condensation? Through experiments that are in a bioarchive paper, we could rule out RBD1 using mutants of RBD1. And in fact, we think this is occurring through RBD2, which again has this interesting overlap with the dimerization domain. And so if we focus in on this domain, which has been shown for other coronaviruses as well as SARS to form a nice dimer here, it's also been seen that temperature can melt this dimer. And what we're seeing is that RNA can melt this dimer as well. And what we're imagining is that double-stranded RNA is essentially acting like a crowbar to pry apart this dimerization domain, and that by lengthening that double-stranded RNA that we can further destabilize this dimer. And this essentially, we imagine, is freeing up the protein to actually engage in larger numbers of multivalent interactions to facilitate phase separation. So in this case, the dimerization or oligomerization domain is actually inhibitory for phase separation in this model. So to test this, we wanted to see if, in fact, N protein in our hands was a dimer, and we could see using mass photometry, indeed it is. So we made a mutant in the RBD2, the second dimerization domain, and indeed the second RNA binding domain that overlaps with the dimerization domain. And indeed, we could see that this shifts the protein population to a much more monomeric pool. And when we look at phase separation of this mutant protein that's much more monomeric, indeed, we can see it much more readily phase separates, both in the absence of RNA and also at very low temperatures. And the presence of double-stranded RNA in the mix does not actually change this phase separation temperature. So, this really suggests to us that it's the combination of double-stranded RNA interacting with this motif that is important for setting the temperature of phase separation. So at temperatures that are greater than about 40 degrees, the dimer can melt just through temperature and increase valence via IDR interactions, we think is promoting condensation. At less than 40 degrees, we propose that double-stranded RNA interactions can pry open that dimer, and that the length of this RNA can actually tune the condensation temperature. And we think this is actually quite relevant in terms of thinking about the life cycle of SARS, both in terms of animal hosts, which undergo hibernation, where they have extreme sweeps in body temperature, but also in the upper airways of human hosts, where we see temperature gradients due to air gradually heating and coming in quite cool. So in this first part of the talk, what I want you to take home is that RNA structure can play an important role as a sticker for phase separation, and in this case is actually working to oppose a dimerization domain that then can facilitate further assembly of higher order interactions. So I next want to focus in on the role of RNA primary sequence stickers. So what might be encoded in the primary sequence for specificity and phase separation, and how this may play a role in N protein actually doing the work of condensing and packaging this gigantic genome. So and in this part of the story, I'm going to be really focusing on this other RNA binding domain. So RNA binding domain 2, we've now characterized to have a double-stranded RNA binding preference. And we think, again, is important for temperature-sensitive phase separation. What is RBD1 contributing? And really, the goal here is to ultimately understand this problem of how the virus is going to find its own genome within the milieu of cellular RNA and package and condense this very large molecule. So our first insight into this came from some early experiments that we did where we took different sequences of the RNA genome. We took RNA from the 5' end, from 2 regions in the middle, a frame shifting region, and what was equivalent to the SARS-1 packaging sequence, and RNA at the 3' end, which encodes the nucleocapsid protein itself. And we asked whether all of these RNAs could equivalently drive phase separation, or if there was some specificity in the sequence. And indeed, immediately we were really surprised and excited to see that RNAs encoding or RNAs at the 5' end and including the nucleocapsid readily promoted phase separation. You can see that in this leftmost column and then this nucleocapsid column. And this is mixing protein and RNA together. Whereas these RNAs in the interior, that we happen to pick, frame shifting region SARS equivalent, actually opposed any minimal phase separation that N protein on its own. It actually appeared to have a solubilizing effect. This is so striking because this 5' end and this frame shifting RNA sequence are exactly the same length. So they have the same charge, the same mass. And so this really speaks to sequence specific differences. To begin to understand what these sequence-specific differences are that might explain this behavior, we teamed up with our collaborators at UNC, Kevin Weeks and Mark Wernicke, to apply a technology they developed called Rnp Map, which allows us to both determine the RNA structure of a target RNA, but also map where a protein cross-links, and the cross-link sites are shown in green here. And this graph, what we're seeing is the primary sequence of the RNA, and each of these green spots, or green lines, I should say, is a point where the N protein is highly enriched or bound to or interacting with the RNA. So what we could see is when we performed Rnp Map on this 5' end RNA that promotes phase separation, we saw a few discrete binding sites, which pointed to us potentially there might be some stickers embedded in these sequences. Interestingly, when we looked at the frame shifting element that limits phase separation, first we could see that this RNA is incredibly highly structured, but we can also see that N protein is making many contacts all throughout this RNA. So it is a very different pattern of interactions. This appears to almost have solubilized the protein. This appears to have the protein bound in discrete spots. So we went on and tried to understand if perhaps these discrete spots were due to RBD1 interactions. So we made a mutant in RBD1 called Y109A. This is a single point mutation in nucleic acid binding pocket. And there were a couple of areas that appeared to be really diminished in interaction in the mutant. And that's just being highlighted here, with this peak being much lower in the mutant than in the wild type protein. And Christine, a super talented postdoc who's been driving all of this work, noticed that in fact, these sites that seem to become less interactive with N protein are actually TRS sites, and that other sites that appear to also be somewhat diminished had a lot of similarity to TRS. These are transcription regulatory sequences that are highly conserved in coronaviruses. And it began to make us think that potentially these sequence elements could be an important sticker for N protein. So we wondered whether these TRS sequences are important for LLPS. And so she went on and made a series of different point mutants in the primary sequence of these elements that appear to be bound by N protein. And indeed, she could actually find very specific point mutants that completely abrogate phase separation and other point mutants that severely limit phase separation. And so we wondered if potentially versions of these primary sequence elements could be potentially found throughout the genome, and that maybe this is going to give us a clue into how RBD1 may be recognizing the genome so that N protein can actually package this large RNA molecule. And so indeed, when we began to look for TRS-like sequences that contain elements of that primary sequence, we could see, in blue here are representative sites where we see the presence of these TRS-like elements. And we can see that they're actually peppered throughout the entire genomic RNA molecule. And so this led us to start thinking that potentially this could be an important sticker for RBD1 for packaging of the genome. And so how we've been thinking about this is, as we think about the fully packaged viral RNA genome, some beautiful electron micrographs have shown this arrangement of Rnp, essentially eggs in a nest type arrangement. So these sort of small globules of RNA and protein arranged within the capsid. And so we've hypothesized that potentially this spatial patterning of TRS-like elements, actually interspersed by these more solubilizing elements, could play a role in this condensation process in ultimately compacting and organizing the genome into these nucleosome sort of like organization. And we've been exploring this through a couple of different approaches. One is using some coarse grain modeling. You've already been introduced to this platform from Rohit. This was developed in his lab, the Lattice platform, where we've been trying to understand how, if we pattern these RNA stickers on beads that represent the RNA genome, if we pattern them in different ways, what does this lead to in terms of the organization, the higher order organization of the RNA? And indeed, when we mimic these TRS-like elements being patterned throughout the genome, what we see is, with a single RNA molecule, which is being represented here in yellow and blue, we see these nice sort of condensed globules of N protein, sort of reminiscent of how you might imagine those eggs assembling. And in fact, if we just look by electron microscopy at actually conditions that are suboptimal, so really subsaturation concentrations of RNA and protein, we see this nice monodispersed, relatively monodispersed assembly of spherical RNA protein clusters, and with a diameter of about 40 nanometers. And this would be consistent with what you would expect if you had 30 kb genome compacted into 30 to 35 Rnp eggs per virion. And this is approximately 1,000 nucleotides per egg. So we think this could be mimicking the packaging unit of the virus. Some interesting questions that this has really led us to think about, though, is, and I think this is also relevant to thinking about chromatin and compaction in the nucleus that we heard about from Cliff, is thinking about whether this is really a 1D or a 2D system, or the RNA sequence, it's clear, is important here. There are surfaces that are really relevant for N protein to be condensing upon, and yet these are the RNA is clearly contributing to the phase separation, but yet it's also essentially acting as a surface. And so I think it's really interesting to think about these sort of size inequalities when we have components in a ternary system like this. And the second thing that's very clear is that something must be preventing coarsening, another theme that keeps coming up in the afternoon talks. And I'll just this has already been talked about, but I'll just highlight this preprint from Rohit and Tony, which we've just heard a lot about, looking at subsaturated solutions and cluster formation, and whether this may be akin to what's going on in that system is a working hypothesis right now. And so what I've talked about so far is really two different kinds of RNA stickers in the genome for N protein. We have RBD1, stickers that appear to mediate LLPS by a primary sequence encoded TRS sequence element, and RBD2 mediating LLPS by interacting specifically with a preference for double-stranded RNA that's promoting and encoding LCST by melting this dimer and competing with this dimer, we think, to enable multivalent interactions to drive larger condensates. So here we've talked about two different modes of encoding a sticker through structure, through primary sequence. I want to just spend the last minute talking about our initial thinking about RNA-RNA interactions and whether RNA-RNA interactions could form a third type of sticker relevant for phase separation. And so I want to highlight here, we've been thinking a lot about these structured elements as I've been showing you models of the RNA, but we see that there's also a fair amount of predicted single-stranded areas or areas that could be available either for single-stranded recognition by proteins, but also for sequence-based RNA-RNA interactions. And so a question we've been thinking a lot about is, are these intervening sequences acting like spacers of some sort? Could they actually modulate the number of spacers that are active at a given time in a system? Or might these actually be creating additional stickers? And so we're still trying to understand this, but we do have ideas for which areas of RNA are tending to be involved in intramolecular interactions. So we've been able to start measuring where we can see single-stranded RNAs have the capacity to interact to form higher order RNA-RNA structures. And in the biology of the virus, we think these kinds of interactions are likely quite relevant to circularization, recombination, transcription. And in all of these steps, this might be a way to actually control the phase behavior of N protein. And so just our initial thinking about this problem, we've identified a few interaction sites between our RNAs of interest that we've been focusing in on at the 5' and the 3' end of the genome. And one of our stickiest interactions, I'm just representing here, it's a short sequence here. And in the next experiment, I'm just going to show you manipulations we've been doing to this potential interaction site. What we've been able to do is essentially recode this interaction site.
Amy, I'm sorry to interrupt. You've got 5 minutes.
Okay, yeah, great. I have my last data slide, so I'll be finished in just a minute. What we can see is by increasing the binding strength between this, by essentially increasing the GC content. So here you can see this is a much more stable interaction. In fact, what we're doing is very much abrogating phase separation. We can also see a different sequence where we make this a longer palindromic-like structure. We also abrogate phase separation. And I want to emphasize this is in the context of a much longer RNA molecule. So we're making very small changes in the potential for this molecule to oligomerize. If we can recode this now back to a more wild type like interaction energy or stability, we can see this restores phase separation behavior. And again, we can actually recode this yet again. And again, the binding energy, the strength of this interaction here in this case opposes phase separation. And so what this is telling us that these trans interactions like this have a really potential powerful role in tuning the phase behavior, and that this might be a way to create different kinds of condensates at different stages of the life cycle. So with that, I'll just summarize and close that hopefully you now have an appreciation of the amazing amount of information that is encoded in the RNA molecule, both in its structure, in its primary sequence, and its ability to oligomerize. I just want to leave you with this message that RNA sequences can control the location, the composition, and the material state, which I didn't have a chance to talk much about, of condensates. And so I hope hope you leave with an appreciation for an amazing amount of information that's present within this molecule that's relevant for phase behavior. So with that, I'll close. Really, all the experiments in this talk were driven by an incredible postdoc in the lab, Christine Rodan, and supported with applied mathematician Ian Syme. I'll just thank my collaborators and my financial supporters, and thank you for your attention.
Thank you very much, Amy. That's terrific. It puts SARS-CoV-2 research into a new and very interesting light for me. I'm looking at the chat for questions, and Jason Kahn asks, can you modulate phase separation with small exogenous RNAs that either block or potentiate interactions?
Yeah, good question. So we've done a lot of experiments also with non-specific RNAs. And so nucleocapsid will absolutely interact with other RNAs in addition to SARS, and that's been well documented by others. There's nice data for that of it interacting with host RNAs. And so indeed, this can absolutely modulate the phase behavior. We see in general, when we've mixed viral RNA with host RNA, that this lowers the concentration of RNA and protein needed for phase separation. It can actually facilitate phase separation. We've had trouble doing experiments where, for example, we use oligonucleotides to block specific binding sites because N protein will bind to DNA as well with lower affinity, but it still will interact with oligonucleotides. So it's a potent nucleic acid binding protein.
Fascinating. Thank you. Thank you very much. I don't see any other questions in the chat. We're right at 2:45. I'd ask you to show your appreciation for our first three speakers, and confirm that we're going to take a 30-minute break and reconvene sharp at 3:15 p.m. Eastern Daylight Time, when we'll hear first from Ned Wingreen from Princeton University. So please come back in 30 minutes, answer your email, take a bio break, and we'll look forward to seeing you again. My thanks to Cliff, to Rohit, to Amy for a terrific start to today's crash course.
There we go.
So we'll resume in a couple of minutes. Ned, are you ready? Great, thank you. Michelle, could you make Tanya Mittag a co-host again, please? She had to step away briefly and lost her status.
Yep, I'll do that right now.
Thank you. Great. Okay, well, it's 3:15 Eastern Daylight Time, and I welcome you back to the second half of today's Institute for Quantitative Biomedicine crash course at Rutgers University, co-sponsored by Princeton University and the Rutgers Cancer Institute of New Jersey. Our next speaker is Dr. Ned Wingreen, who is the Howard A. Pryor Professor of the Life Sciences and Professor of Molecular Biology, and at the Louis Sigler Institute for Integrative Genomics at Princeton University. After undergraduate training in physics at Caltech, Ned received his PhD in condensed matter physics at Cornell as a Hertz Fellow. His title today is The Conformational Entropy of Biopolymers in the Dilute Phase Sculpts Sequence-Dependent Phase Boundaries. Ned, the floor is yours. Thank you.
Thanks so much. Let me share my screen, make sure this is going to work for me.
You're good to go. Thank you.
Awesome. Well, thank you so much for the opportunity to talk today. I've really enjoyed the fantastic talks we've already heard, and they have been a great lead in to some of the things that I'm going to talk about today. Apologies for the overly long title. I promise to keep fewer words on the remaining slides. So what are we talking about? Well, here's my outline. I want to start just with one slide, making a case that you may well be familiar with, which is that phase boundaries depend on a competition between the dense and dilute phases. Clearly that's true. But simple theories, which we often like to write down like Fleury-Huggins, neglect something important, which is the behavior of the polymers in the dilute phase. There are other theories, of course, that do a better job, but I just want to highlight this. And as in many of the talks we've just heard, emphasize the importance of what's going on in that dilute phase. So I'll then talk specifically about the dependence of phase boundaries on sequence of motifs, of specific heterotypic interacting one-to-one motifs. And then I'll segue into what seems like a different story, a somewhat older story from my group about magic numbers shaping the phase boundaries of associative polymers. But I want to make the case that it's really the same story all over again, that you have to pay attention to what's happening in the dilute phase. And then I'm going to conclude. So what about the mean field theories that we love and we teach our students in their introductory classes? Well, Fleury-Huggins mean field theory is always comparing free energy density to some reference state of separating the solvent and the polymers, and then you show them that you imagine now a mixed state where there's a mix of polymers. But in the end, we write down something that looks very nice and simple, that has some contribution from the translational entropy, the entropy of mixing, and then an interaction term having to do with sort of a mean field interaction between the biopolymers themselves and the solvent in itself, and between the two. And we get beautiful free energy density as a function of concentration, and we get two phases, and class is over. But what this doesn't do, and what we don't tell our students about probably, is that this kind of mean field approach ignores the polymer self bonds in the dilute phase. When we show something like this and take a mean field approach, we're ignoring the fact that, well, this polymer might not actually want to be open like that. It might want to collapse looking more like this, or if it has specific binding sites where this monomer is different from this one and it likes to bind to it, it might have all of these different conformations that it would prefer to have, which would then lower the free energy of what's going on in the dilute phase and substantially change this phase boundary. In this case, perhaps shrinking the two-phase coexistence region. Okay, we may have been aware of that, but let's push on and think about the implications of that. And of course, we're not the first ones to think about what's happening to those polymers in the dilute phase. So here's something I just borrowed from a nice paper from the Shea group at Santa Barbara, where they were considering some polymers that have regions of charge. So this polymer here is sort of alternating plus and negative charge, all the way down to this other polymer here, which has long stretch of positive charge followed by a long stretch of negative charge. And by doing simulations, they were able to show that the specific sequence of charges really matters to the behavior of this polymer. So if they're alternating, in this case, they found that the polymer was basically in an open configuration with a large radius of gyration. But if you had these large domains of positive charge and negative charge, they'd have a strong attraction for each other, and the polymer would tend to fold up on itself. So that's true, and it's very clear that that kind of thing can happen with charges. But as we've heard in some of the earlier talks, charges are certainly not the only thing that are holding these condensates together. So I borrowed another nice image from a paper that Rohit is an author on. And this summarizes things kind of nicely. And actually, if you heard his talk, you would have seen pictures that look like this, of this kind of network that is how we understand many of these condensates, where there's specific interactions taking place between residues or other larger motifs on the particular polymer. So here, highlighting what's going on in this little region, you see there's these specific one-to-one interactions between, arginines and, electronic rings here and so on. And so the question, we set out to address a couple of years ago was, well, is this the same story as this charges? But what if the phase separation is caused by this kind of specific short-ranged, one-to-one heterotypic interaction? So in that case, does the sequential order of motifs matter for phase separation? And again, much like this case with charges, we're going to consider polymers where you haven't made more interactions or longer polymers all the same length, all the same number of motifs, same type, just changing the sequence of those motifs. Does that make any difference to the phase separation and the phase boundaries? And the answer is going to be yes. Otherwise, this wouldn't be a very interesting talk. And so I want to spell out exactly what we did, and sort of again following along the lines of Rohit for looking at these thermodynamic properties. It's often very convenient to do things on a lattice. So this is going to be a lattice model for these heterotypic stickers. And in some sense, this is a sticker spacer model where the spacers, we left them out. This is just a sticker model. There's no spacers. So there could have been spacers, but we decided we wanted to just focus on specifically what was happening due to this sequence dependence. And so the idea is, principle, we have a polymer that's on the sites of an FCC lattice, and it has two types of, think of these as motifs or residues. And the only basic rule is that A's like to stick to B's. And in our case, to keep things really simple in terms of the implementation of this, if A likes a B, they actually get together and they sit on the same lattice site. So this polymer here, or some version of it, can accordion up so that A's sit on top of B's. And when that happens, the red and the blue get together to make green, which I realize isn't really what happens when red and blue get together. But it was easier to see green than purple. And the only other rules are that that's the most you can get on any one lattice site is an A and a B. You can't get two A's, two B's, or anything like that. So that the system is in some sense has some self-repulsion. And then to drive phase separation, there's some energy that favors these A's and B's to get together, and also some very relatively weak non-specific interaction that helps give you condensed phases between any neighbors, just as sort of like a van der Waals type of interaction. And so then the only question is, all of these sequences have exactly 12 A's and 12 B's, but the block size is different, going from block size 1 all the way up here to block size 12. And then, we're going to try to see if we can study those phase diagrams, if it matters what the exact sequence is. And then, of course, can we understand why it might matter? So, what do we do? Well, we do Monte Carlo simulations. And in fact, we're using something called grand canonical. And we thank to my colleague at Princeton, Thanos Panagiotopoulos, who's really been the leader and guru in developing these kind of approaches. And the idea here is, we don't want to focus on interfaces between the condensed phase and the dilute phase. That's not what we're about. We really want to know the bulk thermodynamic properties. And so one can do that in the following way. One can just take a box with periodic boundary conditions and then fix the volume of the box and the temperature and vary the chemical potential of the polymers in some external reservoir. And so among the things that can happen is that polymers can appear in the system and disappear. So the number of polymers is not fixed. That's going to be a variable, as is the total energy of the system, which is sort of more familiar when people are doing Monte Carlo simulations. So We're going to propose moves just as in ordinary Monte Carlo, this kind of move and that kind of move. As I told you, there are other moves where we insert and remove polymers. And of course, because of the nature of our interactions, where a red and a blue can get on the same lattice site, we have these kind of accordion moves where within a polymer, a red and a blue can get together on the same site. And of course, all of this is reversible. If they got together, they're able to move apart. And then we just do ordinary Monte Carlo to develop, if you run it long enough, you're guaranteed to get an equilibrium ensemble, in this case, of the number and energy of states. And that sounds great, and that's what we do. It turns out there are all sorts of tricks you need to get this to work, because the system tends to get stuck in metastable states, and and to get close to the critical point, you need to understand that this is in the right universality class. But I don't want to tell you about tricks today. I'm hoping to focus on results. So I'll just show you something, what a result would look like. And so for a particular sequence where we would have block size 3 A's, then 3 B's, we can run this Monte Carlo simulation. And some of the time, what's in the box will look like this. And this is not so far from the critical point. So this is actually the dilute phase. It's not extremely dilute. We can go down to lower temperatures, for example, at which point the dilute phase would be more dilute. But this is very different from the dense phase, which will form in the same box at the same temperature. And the system will fluctuate back and forth between these two states. And so when we keep track of what the system is doing, we'll find this histogram with these two peaks. Sometimes it's down here with 150 polymers distributed in the system, and sometimes it's up here looking like this with more like 900 polymers. And from doing this and from balancing the weight of these two so that we know we're actually right at the point where we've defined a system with equal free energies in the dense and dilute phases, we can extract from the mean of these what the densities of the dense and dilute phases are. And from that, we can then generate the phase diagram that you're sort of waiting to see, with the two-phase region. And then we're going to see that. So here it is. But now for different lengths of these blocks. And so, okay, so, how do the blocks matter? And you could say, well, this has got a suppressed 0 here. These are the tops of the phase diagrams, which we get very accurately this way. And you might say, well, I don't know, that's not so impressive. This went all the way from, a critical temperature in the relevant units of 1.05 to maybe 1.3, not even a factor of 2. On the other hand, we're talking about biomolecular condensates in living systems. So if this guy is at, room temperature at 300 Kelvin, this one phase separates, 60 degrees Celsius lower. And so from a biological point of view, this difference is fairly huge. And so then, okay, so then what do we learn? Well, there seems to be some kind of systematic dependence of this, this critical temperature, and therefore the tendency to phase separate on the block length. And so here we see that indeed for the longer blocks, the critical temperature is higher. That is, they will phase separate more readily. Then there's a kind of systematic behavior here, although you notice that, I hope you can see it, that block size 1 is sort of out of order here. And I can comment on that a little bit later. And we absolutely do understand why it's out of place, and it will make sense. So that's good. So we see that there's some difference, maybe biologically meaningful to the actual sequence. And so of course, this is something that evolution can play around with readily, moving the motifs around within any particular biopolymer. But we'd actually like to understand something about this. So the question is, okay, why do the phase diagrams depend on sequence? And it turns out there's really one dominant factor, at least for this model, and that has to do with what's happening in the dilute phase. What are these polymers able to do when they're all by themselves in the dilute phase? And so what turns out to matter is the entropy of self-bonded conformations. And the idea is I can give you two different sequences that have the same numbers of A's and B's, but then if I leave them alone in the dilute phase, at a regime where the interactions are reasonably strong, so A's do really like to bind to B's at this temperature, then this sequence has lots of options for how to get its A's to bind to its B's. You know, this A can bind to this B, or this A can bind to that B. These A's can bind to those B's. There's just sort of lots of things that can happen, and therefore lots of different conformations that all give you a reasonable amount of self-bonding. By contrast, this one that's all A's and all B's, I mean, it certainly can form self-bonded conformations, but they tend to have a relatively restricted number of them. I mean, it can certainly zipper up like this. That's great. But of course, that's one conformation. There's not a whole lot of entropy associated with that. And, you know, it can have some shifts and so on. But sort of intuitively, it's going to be, you know, more constrained if this polymer wants to form a lot of A and B bonds than if this one does. And so we can quantify that in terms of a density of states. We can actually, in this model, explicitly count for each of these polymers, if it wants to form 5 self bonds, how many different conformations are there on the lattice? If it wants to form 10 self bonds, how many different conformations are there on the lattice? And then, you know, we can just report that, you know, that number as a function of S for all of the different block sizes that we've considered. And so when you do that, you get a fairly striking result that's consistent then with what we see in terms of the critical temperature. That is, the density of states, you know, is substantially higher actually at all numbers of self bonds where you have these small block size. And actually this is block size 2, which has something to do, I should say, with the fact that it's an FCC lattice. But like, don't worry about the fact that there's details of the lattice. The important part is that this order of the density of states, the more things, the more ways you can form self bonds, the less likely you are to want to phase separate. And that's, you know, that clearly is dominating in our system. So it's really, even though, yes, there are differences in the dense phase because these are polymers with different sequences, they have different ways of organizing themselves, forming trans bonds in the dense phase. The thing that dominates the critical temperature is actually this entropy of self-bonding in the dilute phase when it's just individual polymers floating around. And so you can find that this also has implications for the critical density at which things form. This is actually a fairly straightforward result that sequences don't condense until, you know, trans bonds become favorable over those self bonds. And for the lower TC sequences, the ones that are less or more reluctant to phase separate, this is going to happen at a higher density. Kind of maybe more interesting is something about the behavior of the polymers in the dense phase, having to do with how many trans bonds they have. Now, we would love to actually tell you in this lattice model what the viscosity is, but the problem is if you're studying a lattice model, you know, there really is no viscosity. On the other hand, we can be guided by polymer physics to report on what the viscosity should be based on the number of connections between polymers, because basically what's limiting the diffusion of polymers in the dense phase and limiting the overall viscosity of this material, say in response to shear forces, is the ability of a polymer to break bonds with other polymers and then move apart. And so really, all we need to do is count trans bonds. And that's something we definitely can do in these Monte Carlo lattice models. So as long as we believe that the dynamics are dominated by the trans bonds, and we can thank Rubinstein and Semenoff for some beautiful analysis along these lines, then we should be able to make good predictions for the viscosity and diffusion. And what we learn is that polymers with large blocks have slower dynamics. And that's because it turns out that just like in the dilute phases, it's hard for these guys that are like 12 A's and 12 B's to form self bonds. And so also in the dense phase, they tend to form more trans bonds. And with those more trans bonds, the whole dense phase is not actually more dense, but it's just less dynamic. It has to break more bonds in order for the polymers to diffuse or to move around to give rise to that gives rise to the viscosity. So here you see the important ratio is that the diffusion coefficient depends inversely on the number of trans bonds per polymer. And actually for the viscosity, it's even stronger. The viscosity should scale like the number of trans bonds squared. So it's really quite a strong effect depending on the number of trans bonds, which depends, and again, on this detailed order of the motifs. So I am happy to report that I have a little bit of time, so I do get to tell you about this other story, which again, I'm going to relate back to this one as follows. So this is some story about magic numbers. I'll explain to you in just a moment what I mean by that. But this is some work that was done by Yaojin Zhang. She was at Princeton. She's now running her own group at Johns Hopkins. And this is an off-lattice study, but again, with these heterotypic one-to-one stickers. So A's like to stick to B's. And this time, instead of mixing up A's and B's on a single polymer, we're going to consider a case. This is like the polysumo system that Mike Rosen made famous, where this polymer is a poly blue sphere, and this one is a poly red sphere, and they like to interact with each other. And of course, one thing that can happen is that they can form a condensate where one blue polymer interacts with many red polymers and vice versa. But again, it's important to think about what's happening in the dilute phase. They can also form happy couples where red polymer and a blue polymer overlap with each other and satisfy all of their bonds. And that's what's competing with this condensed phase. So, again, not going through too many details, you have to, make choices about the energies of the linkers and the interactions between A's and B's and so on. And then, we use this very popular LAMPS platform for doing Langevin simulations. And then you get things that look like this. You can get a dense phase here, with a dilute phase surrounding it. All of this has periodic boundary conditions. You can look at the densities of these two phases. But what's interesting is that, you want a phase diagram. And now because there's two different polymers, the phase diagram lives in this two-dimensional space. You have a dense phase, you have dilute phases. But then there's this weird thing that happens where, you see, why is there some line in the middle there? And that turns out to be this magic number effect. And the idea is, it really matters whether the couple is happy in the dilute phase, whether the A polymer and the B polymer can satisfy each other. And you can see something like that here. This is a case where we've considered a polymer of length 14, say the red polymer, and a blue polymer with different lengths, different possible lengths. And at relatively weak interaction energies, you look at the dilute phase, and all you see is the dilute concentration is going down as the valence of the B polymer is going up. And you're like, well, that's not surprising. Longer polymers will tend to phase separate better, will have a lower dilute phase concentration. But if you increase the interaction energy, something weird starts to happen. And it happens, by here, you see something sort of very strange, until you figure it out. You have very, very low dilute phase concentration, so strong tendency to phase separate when you have a mismatch between the lengths of the A and B polymers. But then once you get a length 14A and a length 14B, the dilute phase concentration actually is non-monotonic. It comes back up again. And this is just reflecting the fact that these are now happy couples, that they're both length 14 and they completely satisfy each other. And therefore they don't need to go to parties. They don't need to interact with any other happy couples. They just hang out, and the dilute phase doesn't have to phase separate. And you can see that you can form more complicated families, for example, with two length 7s will also satisfy a length 14. You can see it here, not length 14, that was too hard to draw, but you can see that you get these happy families that are now competing with the dense phase. And so again, the theme is, the phase boundaries depend on the competition between what's happening in the dilute phase and the dense phase. So we've really got to pay attention to the free energy density of the dilute phase, not just the dense phase. The work we did with Jun Zhang shows this is true for different combinations here, but also realized that you can change not just the relative valence, but also the overall concentration of stickers. All of this was for a 1-to-1 ratio. And when we started changing the concentration of stickers, we realized that this kind of magic number condition is really when, what it really has to do with the overall relative stoichiometry of stickers as well as polymers, such that what really is being favored here is an equal stoichiometry of the polymers. Because in this case, this is really a low energy state. There's an available bond here, but the whole system has available gold stickers. So there's no overall free energy cost of forming this kind of family. So in the end, you see the magic ratios are suppressing phase separation. So the point of this, showing these two things together, is that in both cases, what's really can control the phase diagram is the properties of the polymers or oligomers in the dilute phase. And so if there's any take-home message from this, it's pay attention to what's happening in the dilute phase. And so with that, I'll conclude. When phase separation is driven by these one-to-one heterotypic interactions, the motif sequence governs the phase diagram through this entropy of self-bonding, whereas the sequence also influences the condensate properties, but this case through formation of trans bonds. And these, of course, are related. For associative polymers, the relative valence and stoichiometry shape the phase diagram via magic numbers, again, depending on how happy are the oligomers in the dilute phase. And that just in general, the existence of these strong specific one-to-one bonds does allow for some kind of biological control, because changing one site on a polymer can have a big effect on phase properties. So if you're interested, there are some references on this. I want to thank the people who did the work. In this, I talked mainly about work from Ben Weiner, who's now a postdoctoral fellow at Rockefeller. And as I said, June Zhang is running her group at Johns Hopkins, and various colleagues, including our own Cliff Brangwyn. So thanks very much.
Thank you very much, Ned. That was terrific. If there are any questions, please put them into the chat. Christina Robb remarks, the first part of your talk reminds me a lot of net charge per residue and patterning of opposite charges in IDPs reported by Papu. Do you think these two things are related to phase separation?
Oh, absolutely. I mean, I think, again, we're far from the first ones to make the point that motif patterns matter. There's been a lot of work in this in terms of charged motifs, and these long blocks of charge are very attractive in a kind of not exactly one-to-one way, but with some parallels. There are some differences here in that in that case there is definitely a sequence dependence, but it's essentially a statement that about energetics, that these large blocks can form a very low energy state by coming together. Large charge blocks can form a low energy state by coming together and phase separating. Whereas in this case of these specific one-to-one interactions, it's not so much a statement about energy as about entropy. So whereas some of the conclusions for the behavior are the same, the driving forces are somewhat different.
And Greg Kretschler, who's a member of the Protein Data Bank team here at Rutgers, asked, you showed a two-state equilibrium with stickers of different lengths. How would adding spacers affect the equilibrium, or different types of spacers?
Yeah, absolutely. And I think, Rohit is the master of the spacer. And, he's made a great case that it absolutely depends whether the spacers are just taking up space, which will disfavor phase separation because the system is more crowded. It's entropically unfavorable to put these things that don't really want to be inside the condensate to keep them in there, or if the spacers lead to some kind of effective attraction, if they have some perhaps weak non-specific interaction, they could favor phase separation. So it is. Yeah, the question of what spacers do really depends on details of the of the chemistry of the spacers, but they do play, they certainly can play a huge role.
Thank you. So with that, Ned, I'll thank you and call on our next speaker, Dr. Carla Matos, who's a professor and director of graduate studies in the Department of Chemistry and Chemical Biology at Northeastern University in Boston. Carla received her PhD in structural enzymology from the Massachusetts Institute of Technology in Cambridge, where we worked in the same laboratory under the direction of Greg Petsko. Her title today is Building a Molecular Model of RAS-RAF Signaling in the Control of Cell Proliferation.
Thank
Carla,
you.
your slides are visible and you're good to go.
Excellent. Thank you very much. I am learning so much by being here. And today I'm going to tell you about a condensate model that we were led to by studying RAS at its molecular details. And I'm going to tell you how we got to that. Of course, it's still a proposed model that we are working to validate. RAS is a master signaling protein. It's a series of 3 isoforms, KRAS, NRAS, and HRAS, all of which function through the classic GTPase cycle, where RAS is off when bound to GDP and on when bound to GTP. And these 2 states of the switch are regulated by GAPs, the GTPase activating proteins, and the guanine nucleotide exchange factors. RAS interacts with a variety of different effector proteins to signal through, to affect a variety of cell pathways that, when usurped and mutated, are associated with cancers. So, you know, the proliferative pathway involving the RAF-MEK-ERK pathway and anti-apoptotic pathways, cytoskeleton and cell migration, all of these pathways are misregulated in cancers. And it's not so surprising that RAS is involved in about 20% of all human cancers, with KRAS being the most prominently mutated isoform of RAS. So having said that, we have not been successful in targeting RAS against cancers, and our lab has been really focused in understanding molecular mechanisms of signaling through RAS, particularly RAS-RAF, so that we can perhaps find new and more effective ways to target this protein. So everything that I'm going to tell you about from now on is really having to do with the wild type protein. So RAS is post-translationally modified at its C terminus, that then it's lipidated and inserted into the membrane, and the G domain then is kind of hanging off the membrane in the cytoplasm. And the structural work is normally done with a truncated protein that involves the G domain. The G domain is composed of 6 beta strands flanked by 5 alpha helices and 10 connecting loops. And the switch 1 and switch 2 regions are important sites of protein-protein interactions, both with effector and regulatory proteins. And this happens to be intrinsically disordered. Both switch 1 and switch 2 have been shown by NMR to be completely disordered outside of these protein-protein interactions, or they're often stabilized in crystal structures by crystal contacts. So here is an example of, you know, how this area, switch 1 and switch 2, gets slightly differently modulated depending on its interacting partners. So let's jump into the work that we've been doing. For many years, we've studied allosteric networks on RAS. And I want to tell you about 2 particular networks. One that we think is linked to hydrolysis of GTP in the presence of RAF. And that's this network here that involves the allosteric site, which ends up ordering the active site and key catalytic residues. And the other one is the a network that links switch 1 to the alpha 5 and its partner alpha 4 helices, which are at a dimer interface, which I will introduce to you throughout this talk. And we think that this dimer interface is key in forming these polymeric platforms that we think and we're proposing is the basis for signaling through RAS. So we discovered this allosteric site many years ago and published now over 10 years ago, when instead of using calcium chloride to crystallize RAS, one of my students used calcium acetate, and we surprisingly found a movement in helix 3 here, which in this yellow structure is facing switch 2, causing steric hindrance and keeping this region disordered. And now, but with bound calcium acetate in this allosteric site, the helix moves back towards helix 4, giving room for full ordering of this active site. And we propose today, but at that time I had no idea what this was all about, but just to give you a little heads up, we think that the acetate molecule is probably mimicking some negatively charged phospholipid head group in actually the cell, which is our hypothesis right now. So the fully autoreactive site in this 1.3 angstrom resolution crystal structure shows an active site with a catalytic water molecule poised for a nucleophilic attack on the gamma phosphate. And this bridging water molecule donated protons, and of course we don't see the protons here, but this is our hypothesis, donated protons to both the tyrosine 32 and the glutamine 61. And we have shown in this paper down here, published in 2015, that the gamma phosphate is protonated. And in our hypothesis, it would be donating this hydrogen bond to the water molecule, which now has a partial positive charge sitting right over what would be in GTP, the bridging oxygen between the gamma and beta phosphates. And it has been known for many years now that the dissociative mechanism would accumulate negative charge on that bridging oxygen, thereby putting a partial positive charge there would help stabilize the transition state of the reaction and in fact increase the rates. So we've done a lot of work on that I'm not going to show here because I want to just give you the highlights of what we're doing. But at that time we thought, oh, this is really interesting. Is this allosteric site specific for calcium over magnesium, for example? So we took our crystals grown in calcium acetate and we soaked in stabilization conditions containing calcium acetate, collected data set, and there it is, calcium and acetate bound. We did the same thing, but now with magnesium instead of calcium. So we soaked in magnesium acetate and there's no binding of either magnesium or acetate. And then we took those crystals and soaked them back in calcium acetate. And there we go, we have our density again. And from that paper, from those experiments, we concluded that this site was specific for calcium over magnesium. That was all very exciting. But 2 years later, we saw this paper by actually colleagues of ours that had decided to characterize this second ion binding site, the allosteric site, using NMR, and they found that divalent ions bind RAS in solution with millimolar affinity, very weak binding, and no selectivity at all, including between calcium and magnesium. So we were fairly puzzled at that time, but we also noted that in the crystal, as opposed to in solution where RAS is strictly monomeric, in the crystals, we could generate a dimer through alpha 4 and alpha 5 here, generate this by a twofold crystallographic symmetry. And that this dimer interface is adjacent to the allosteric site, and that could be stabilizing interactions that make the site specific for calcium in the crystals, right? So, well, what does this have to do with, real signaling in the cell? At that time, we didn't know, but it was something that we kept in mind. And so now I want to introduce RAF, okay, one of its effector proteins. And, the focus of this talk is signaling through the RAS-RAF-MEK-ERK pathway. And so RAF is has nearly 650 amino acid residues. And it has 2 RAS binding domains towards its N-terminus, the RBD, which is a high affinity nanomolar affinity RAS binding domain, and a weaker binding in the micromolar range, cysteine-rich domain. And our lab in 2015 published the crystal structure between RAS and RAF-RBD. And just last year, we published a crystal structure containing both domains, HRAS in complex with the RBD-CRD construct of RAF. Almost simultaneously, the RAS initiative people at the NCI published the same structure, but with KRAS in complex with RBD-CRD, and they are nearly identical. So that's really good to know that the 2 isoforms are actually interacting with RAF in precisely the same way. Interestingly, our crystal structure of the RAS-RAF-RBD complex also had a dimer generated by twofold crystallographic symmetry. This is a completely different space group, actually different contents, because this has RAF-RBD. So that was really interesting. But the dimer interface is essentially identical to the one that we saw in our crystals of RAS alone. And these conditions do have calcium acetate, and we have a calcium acetate bound in the allosteric site in this complex. Furthermore, the structure of the KRAS RAF-RBD CRD also contains the same dimer. And again, different crystal form, different contents, because now we have the RBD CRD. So at that point, could this actually be a real biologically significant interface, and what's happening there, right? But we didn't have any further answers to that at that stage. But however, let me point out the interface between RAS and RAF-RBD from our crystal structure published in 2015, and it's a highly charged interface with positively charged RAF-RBD, negatively charged switch 1, which remember is completely disordered except when bound to this binding partner or stabilized by crystal contacts, as in our structure. And amazingly, the crystal contacts stabilize switch 1 to the exact same conformation seen in our complex between RAS and RAF. And we see the same structure in the active site with the catalytic water, the bridging water molecule, etc. So we were very, very excited. And at that point, we started posing the hypothesis that maybe this is a catalytic mechanism that is facilitated by the interaction with RAF. So a RAF-specific catalytic mechanism that would not involve gaps. Actually, RAF is nanomolar affinity, GAPS is micromolar affinity, so it would make sense that it would have its own hydrolysis mechanism. Okay, so now let's switch to this whole question of the dimer. Right, we have our dimer in the crystal, and interestingly, for simultaneously as we were doing all that work, papers started to come out. Several papers, I've only chosen 2 here, which I think for me was very impactful and really started to change our thinking about all of this. And this has to do with dimers of KRAS being observed in cells in this first paper here. Where they observed by PALMs, which is a photoactivated localization microscopy, they would actually observe RAS dimers in live cells. So that was really, really exciting. Remember, previously, the thinking was RAS is kind of a monomeric signaling pathway. And then in 2018, there was a study that mutated this dimer interface and showed that in cells where the dimer interface is intact, there are tumor cells that proliferate extensively, and that this mutant that disrupted that dimer interface, specific, the alpha-4, alpha-5 dimer interface, really decreased these tumor cell growths. And then they put it in mice and showed that tumors retracted in this mutated version of RAS. So that was also very exciting, validating this alpha-4, alpha-5 dimer that we were seeing in our crystals. Simultaneously, at that point, we decided that we would investigate a higher molecular weight peak that we routinely see in our purification of the RAS-RAF complex. And normally in dilute solutions in this purification, this major peak here of the RAS-RAF complex is dominant, and we always saw a tiny little shoulder here at this point. And what we did is we went to very high concentration, asking the question, if we really go to 50 mgs per mil of this protein or higher, can we shift equilibrium to this higher molecular weight peak? And the answer was yes. In gels, we knew that the contents of the peak was RAS and RAF. And therefore, this hypothesis that it might be an oligomeric form. And indeed, in collaboration with our colleague Lee Makowski here at Northeastern, we collected SACS data right off the SEC column. So it's called SEC-SACS experiments. And we were able to calculate molecular envelopes for each of the 3 peaks. And indeed, it fits the dimer in our crystal structure. beautifully, that one generated by the twofold crystallographic symmetry, the monomer, and this is excess RAF-RBD. So we were really excited about this, and we started thinking, well, RAF may be promoting dimerization of RAS. And around that time, I happened to be at a symposium where Jay Groves from UC Berkeley was showing his data of RAS on supported lipid bilayers, where they saw that KRAS by itself at very high densities, up to 2,000 KRAS molecules per micrometer squared, was strictly monomeric. And in fact, he had published a paper really kind of going against this idea that RAS dimerizes on the membrane, because they had shown that it actually doesn't. This is diffusion that is associated with the monomeric form of KRAS. And I suggested to him that he might want to repeat these experiments with RAF-RBD. And the results were striking. What they found was that at very low surface density, tens of KRAS molecules per micrometer squared, in the presence of RAF-RBD on supported lipid bilayers, they could detect by fluorescence correlation spectroscopy the diffusion associated with a dimeric form of the complex. So that was very exciting. They also confirmed this by single molecule tracking. And we included this, their data in our paper, because this was evidence that RAF really is key in the dimerization of RAS. So now we're linking RAF in the crystal. We saw, that same kind of allosteric site in the presence of RAF that we saw before. And we are now in the bilayer, we're also seeing the effect of RAF in the dimerization of RAS. So we're now proposing a model where, yes, gaps and gaps are still important in modulating the levels of RAS GTP and perhaps in other signaling pathways. But in the RAS-RAF complex, once we bind RAF, we form a dimer, and this dimer stabilizes this calcium binding site and right up against the membrane, where a negatively charged membrane phospholipid group may also be playing a role in the hydrolysis of GTP. So at that point, we thought this is really, really interesting, and we had been doing some molecular dynamic simulations with RAS and the RAS complex, but not yet in the dimer. And what we find, we use for analysis something called dynamic network analysis, which uses the molecular dynamic simulations to look at the proximity of residues within the simulation time, like which residues are within 4.5 angstroms for at least 75% of the simulation times get put together, linked in these communities. So look at RAS has a lot of motion. It's a very dynamic, in the GTP bound form, very dynamic. And it moves with different pieces kind of uncorrelated with each other. These dynamic network analysis are looking at allosteric connections between different parts of the protein. We also can look at pathways of allosteric connections, as shown here. This is for the allosteric site. And what we see is for RAF, again, lots of different communities, very dynamic proteins. And when the complex is formed, look, we have a RAF gelled into a single community, two actually communities, with the loop 4 having its own community. But the number of communities in RAS also decrease. And remarkably, in the dimer, what we see is 2 sets of communities for RAS, one involving the allosteric site. This is the allosteric site here, and that's glutamine 61 that gets ordered in the active site by binding of calcium acetate. So that forms its own community. And the other major community interacts with RAF, which now is completely in one community of residues. So very, very interesting. We were surprised by this. And we also noted that there were thousands of paths, like 25, 30,000 paths that connected these two ends, specifically residue D113 at the opposite ends of the dimer. D113 is a amino acid residue that have been shown to affect the free energy of binding between RAS and RAF. And look at it, it's remote from the binding interface. So we thought it was a really interesting residue to look at when we were doing our path analysis for these allosteric networks. So actually this was determined by 2 independent studies, simulation studies, and they're described here in these references. Not only that, but really, really intriguing to us was the fact that residue 113 was proposed in a model of the RAF interaction with a scaffold protein, galectin-1. Galectin is absolutely required for signaling through RAS-RAF pathway. And interestingly, this residue D113 is proposed and validated by mutagenesis experiments, proposed to be in the binding site between RAF-RBD and galectin-1, which itself is a dimer. So putting all of this together, we really started to think that the purpose of RAS dimerization is not necessarily to induce RAF dimerization, although it may be involved in that in some way, but that the binding of RAF-RBD to RAS GTP promotes a dimerization interaction that modulates, this is our hypothesis, modulates the binding site for galectin through those allosteric networks, thereby recruiting galectin to our proposed platform, signaling platform, which is really a polymer of RAS-RAF and galectin dimers. And these that may be forming condensates, which have been observed, as I'll show you in a minute. And so this is really exciting. It's a completely new model for signaling through RAF, and it's still being validated and questioned. And so I just wanted to make that clear. We're still working on validating this model, doing both biochemical and structural biology experiments to get a complex that includes galectin, the RAS-RAF and galectin trimer. So meanwhile, it's interesting, the role of the CRD. The CRD, really for years and years has been proposed to be inserted in the membrane, because this region here of the CRD contains some arginine and hydrophobic residues that interact, that are actually found in vitro to insert in the membrane, right? So there's that question. And all models so far have assumed that interaction to be absolutely necessary. In this model, what we find is the CRD interacting with loop 8 directly adjacent to the active site. And in preliminary simulation models, we see diverting of some of the allosteric pathways through RAF-RBD, creating some asymmetry in the dimer that are very, very interesting, because the kinase domain of RAF also has been seen to function asymmetrically. So we're still investigating that part. But that really brings a new role for the CRD. And just to summarize our current model, we think that RAS GTP binds RAF-RBD, and thus for activating, releasing autoinhibition of the kinase domain, which I haven't mentioned, but it's part of the whole story. At the same time, this allosteric site sits right here. And without any calcium, then this forms a signaling platform that involves both RBD and CRD modulating these allosteric pathways, recruiting galectin to the proposed galectin binding site. And that when calcium binds at the membrane with a negatively charged phospholipid head group, that activates hydrolysis to dismantle this whole platform. And the really great thing about this platform is that it has the potential of both amplifying the signal and kinetic proofreading, so you don't have spurious signaling of just kind of a unit of RAS-RAF, right? So this actually is really exciting. And the most exciting things of all is that a paper in 2019 actually observed 3 different phases for diffusion of RAS on cell membranes. And they determined, and here are the different rates of diffusion: a fast diffusion rate, one that is intermediate, and an immobile phase. And we propose that the immobile phase consists of these condensates made of these polymeric signaling platforms that we deduced from our models and the work on allosteric pathways that we've been involved in, and that this neutral phase, this blue phase, is kind of an intermediate phase that has oligomeric, very, RAS molecules that are kind of close to each other, interacting weakly through various oligomeric interfaces, get ready to form the immobile phase. And with that, I want to finish and thank the people who in my lab who's done this work. Jillian Parker started the whole dimerization project and is just finished by Morgan Plock. Trinity Cookis did the structure of the RBD-CRD that we recently published. Various people in the lab have been involved with a lot of different aspects of this project. And I want to thank collaborators in Jay Groves and his two postdocs, Lee Makowski and his graduate student with whom we collaborated on this project. Thank you very much. And if there's time for questions, I'd like to take some.
Thank you very much, Carla, for a great talk. Yes, there is time for a few questions if people would like to put them in the chat. While members of the audience are typing in their questions, Carla, I wanted to ask you a crystallographic 3D structural question about the buried surface area that forms when RAS dimerizes.
Yes. The buried, I don't remember the exact surface area, although I've looked at it before. It's substantial. It, you can, it's this entire.
It does look fairly hefty. So my suspicion is that that's going to be large enough that you might think that it would occur in solution, and you've got some evidence from the small angle scattering data to back that up, don't you?
Yes, but only in the presence of RAF, right? So RAF kind of stabilizes, and we've shown that binding RAF affects the dynamics in the allosteric side and also the helix 4 helix 5 dimer interface. We've shown allosteric pathways leading from the RAF RBD binding site to this dimer interface. So I think RAF RBD is a key feature in forming this platform.
So this looks like, and I think that Greg Critchlow from Protein Data Bank is alluding to this in his comment. I think he's already been on the PDV website, rcsb.org, to look at the structure, and commented that there's a potential of a conformational switch that interferes with dimerization until RAF helps to induce the conformation which is suitable for forming the dimer. So this must be balanced on a knife edge.
Yes, I think so.
In terms of this equilibrium between monomer and dimer.
I think it's very finely controlled. Not only that, our hypothesis, in which we're now investigating the interaction between RAF, which occurs over here, right at this end of RAF, or both ends, and galectin, because our hypothesis is that because we see these allosteric modulations, a very significant change in the dynamics in this region, and we think that modulates, probably pre-arranges the binding site for galectin, which does not interact outside of this dimer form, right? So the whole dimer is priming the platform, the recruitment of galectin and formation of this signaling platform that we're proposing.
So I'm going to take the chair's prerogative, if you don't mind, Carla, and ask you one more question. Is there any, have you entertained the possibility that that the findings that you're making here would identify a potentially a second druggable site on the surface of RAS?
Yes, and the most obvious one to me would be because RAF RBD is actually very, very dynamic and weak binding. And I think that targeting that site around this area might be very effective. You know, there's the whole allosteric, the allosteric site, although you have to be careful about that because that's actually controlling hydrolysis, and you don't want to mess with that too much. But there's the potential of drugging RAS in the presence of RAF-RBD, and very few people are doing that out there right now.
Yeah.
And I think
there's one approved drug for RAS, and that's
Yes,
the
there is
that's the
one.
G12C mutant form of
Exactly.
the protein. Yeah.
exactly.
Well, with that, Carla, thank you very much for a great talk, and we'll now move on
Thank
to
you.
our final speaker of the day, Dr. Tanya Mittag. She's a member of the structural biology program at St. Jude's Children's Research Hospital in Memphis, Tennessee. Tanya received her PhD from the Goethe Institute in Frankfurt, Germany, and began her work on IDPs as a postdoctoral fellow at Sick Kids Hospital in Toronto, working with Julie Foreman-Kay, who also worked in the PETSCO lab in the same time that Carla and I were there. So Tanya's title today is "How is phase behavior encoded in prion-like low complexity domains?"
Thank
Carla,
you
the
very
floor
much,
is
David.
yours, and you've just go into presenter mode, please.
Am I on the, do you see the wrong display?
Sorry, you're not yet in presenter mode.
I should be, actually. So do you see, you see the
I
slide?
see a
and the
line of
slide?
slides. I see a line of slides on the left of the screen.
I
You
have
aren't
switched
getting
a
into
couple
full
of times
screen.
now. Is this working now? Is this better?
No, you're still not
Oh.
there. We're still seeing the.
That's really strange because I'm
yeah.
swapping displays. Give me 1 second. My
Okay,
apologies.
this is the multiple display tyranny.
Yep.
Yeah. Bear with us, everybody, please.
Right.
You've still got the same challenge.
Okay. How about now?
It still looks the same.
This
Sorry.
is so weird. I'm
Yeah.
switching all the time.
Yeah. Well, I suggest that you start and go with the flow. The floor is
Okay,
yours.
my apologies. I don't know because I'm switching display, so this should fix it, but I don't know why it's not, and I see it switching on my side. All right, I will speak anyway, and I'll tell you about how phase behavior is encoded in prion-like low complexity domains, which is one of the interests in my lab, and I'll focus on proteins and stress granules. Because stress granules are an important type of biomolecular condensate in the cytoplasm. They form after acute stress. They are very dynamic. They fuse and coalesce, as you will see here in this nice video from my colleague Paul Taylor. And their formation is reversible, as you'll see at the end of the movie when the stress condition is washed out and they disassemble again. And these stress granules form when translation is stopped upon stress, and
I don't see any. I don't see the movies. I'm not sure if I'm the only one.
okay, I am sorry. I need to stop sharing again and reshare. I don't know what the problem is. What do people see now? The full
Now we see
screen
your
with the movie?
notes.
And now we can see your notes and
can
the
see your
line
notes.
of thumbnails on the left.
Yeah.
Actually, it doesn't fill the full screen.
Yeah, and it doesn't fill the full screen either.
I have never had this problem before. I've given lots of talks
Tanya,
online.
Tanya, maybe you can show your full screen versus just the presentation window.
Yes,
This
I
is
can
good.
try that.
Perfect. Yep. Yep. For a moment you had it.
Well, I stopped sharing for a moment, so
Uh-huh.
I don't know why it worked.
Okay, we can now see the full screen.
You can, because I'm not even in presentation mode right now.
No.
Okay.
Yeah. So we can at least see the entire slide at this point. And now we can't see the entire slide once you go into present mode, and we're still seeing your notes. Ah, this is it. No, okay. I don't know what.
That's when I'm leaving, and I
Only
can't.
when you're leaving is it working? Perhaps
Okay,
you could turn off
my whole
turn off your
desktop.
second display. Is that possible?
Yeah, that should be possible. All right, let's try this. How is this working now? Do you see something now? You see the full?
We're not seeing anything now.
Really apologize. Okay, I hope now I have it.
You have it.
Okay, good. No more two displays. All
You're
right.
good. Yep.
I apologize. I'll probably jump ahead a little bit and say we're looking at stress granule proteins, and that's important because these stress granules, are important for the cells, but also a lot of the proteins inside stress granules can lead to the formation of solid fibrillar inclusions and people with neurodegenerative diseases such as amyotrophic lateral sclerosis and frontotemporal dementia. And there are familial forms of these diseases where mutations are found in RNA binding proteins that go into these stress granules and are major components of these stress granules. And so these mutations lead to favoring the formation of these solid fibrillar inclusions, basically husks of stress granules, or other mutations in ubiquitin-dependent autophagy factors, stress granule disassembly factors. And if these factors are not active anymore, that also favors these disease processes. And so there's this very strong genetic link between stress granules and development of the diseases, and the fact that a prolonged assembly of stress granules lead to the diseases. And we showed a while ago with my colleague Paul Taylor that proteins, RNA binding proteins that are found in stress granules can undergo phase separation. Here we have wild type protein in green and disease mutant protein in red. They both form condensates in vitro. And the disease mutant protein, it has a mutation that leads to enhanced fibrillization of the protein. It fibrilizes strongly from within the condensates, as you can see here in the movie, where these droplets settle on the glass slide and deposit their fibril load, the fibrils that have formed within them. And so phase separation really enhances fibrillization of these proteins. And this kind of explains why stress granule, or is a possible mechanism why stress granules are crucibles of these maturation processes that lead to neurodegenerative diseases. So RNA binding proteins with low complexity domains, the type of domains that I'm going to talk about, are important for physiological functions in cells, for the formation of stress granules, but also for development of different diseases. Neurodegenerative diseases, as I showed you, there's fibrillization going on and the deposition of insoluble protein deposits, and also in cancer, because the intrinsically disordered low complexity domains of RNA binding proteins often get fused to other parts of proteins, for example, to DNA binding domains of transcription factors, and they drive cancers by inducing aberrant transcriptional programs. So that's why we think it's important to understand the phase behavior of these proteins. And the protein that I'll focus on is HNRNPA1, an RNA binding protein that is found in stress granules. It shares a lot of the features of these typical RNA binding proteins, including two-folded RNA binding domains called RNA recognition motifs, and this long intrinsically disordered so-called low complexity domain that is sufficient for mediating phase separation. And it's called low complexity domain because it doesn't use the full amino acid alphabet. About 60% of the sequence are made up of lysines and serines. There are lots of other small polar residues, and then we have some aromatic residues and some charged residues, and they'll become important again. But overall, these are sequence features that are shared with a lot of other prion-like low complexity domains and RNA binding proteins, but also in general with other phase separating proteins. And so this is a relevant system to look at in more detail. We've heard a lot about how phase separation is mediated by multivalent interactions. A reminder here of a multivalent domain motif system like the SH3 proline-rich motif system that was first shown to how it phase separates by Mike Rosen in a seminal paper in 2012. We have these multivalent interactions. They can lead to the formation of large non-covalently networked complexes. And if these are relatively insoluble, they undergo phase separation and you have a networked dense phase and a coexisting dilute phase with these clusters that Rohit spoke about. But then some intrinsically disordered regions can undergo phase separation as well. And how is this encoded in their sequence? Well, if we knew their interaction motifs and interaction strengths, and I think that this should work very similar to domain motif type systems, where we understand very well that the valence and the interaction strength determines the phase diagram. You see here a schematic phase diagram. If we change the valence of protein A versus protein B, both are multivalent for each other, then that changes the coexistence line here, the line above which we would first get phase separation. So if the valence becomes higher, these proteins can network more easily with each other and can lead to the formation of a network dense phase at lower protein concentration. So if we now go to these intrinsically disordered proteins and say, if we knew the interacting motifs within them, then we would expect that a higher valence or higher interaction strength of these motifs should also lead to stronger phase separation in these intrinsically disordered regions. And here is our schematic phase diagrams that would show this, where we have concentration, of course, only of one protein, and then on the other axis, some sort of interaction parameter that could be changed by pH or temperature or salt condition. And so to address this question, we're using the stickers and spacers framework that was introduced by Rohit Papu today, but also by him into the phase separation field from studies of associative polymers, where stickers are adhesive elements that form physical crosslinks with each other and mediate networking of the proteins. And spacers connect these stickers and influence their ability to interact with each other. All right. Rohit already told you that aromatic residues are really important in these types of sequences. And we decided that we'd be able to find the interacting motifs in individual proteins. We didn't actually have to go to the dense phase, because the same interactions that would drive phase separation intermolecularly should also be satisfied intramolecularly and compact the protein molecules. And our data, which I'm not going to show you because it's been published a while, really pointed to the importance of aromatic residues in the sequence that I've highlighted over here. And we made a bunch of mutations in which we changed the number of these aromatic residues and measured the single chain dimensions of these proteins and really saw that the single chain dimensions changed. And this allowed Alex and Rohit to extract interaction strength, sticker interactions, but also sticker spacer and spacer interactions. That then allowed them to do stickers and spacers, lattice simulations, where they put a number of chains into a box and simulated their behavior with the interaction strength that they had extracted from our experimental data. And that allows the determination of full phase diagrams. And these fit our experimental data here as triangles, in fact, very well. So that means that the aromatic residues are really important for this phase behavior, right? Because we were able to recapitulate the phase behavior with these parameters. What it, of course, also showed was indeed that the valence and the interaction strength of the stickers, in this case, the aromatic residues, determines the phase behavior of these low complexity domains as we expected. So if we go to this ARA plus variant, which has a larger number of aromatic residues than the wild type here, then we need a lower protein concentration to first hit the saturated, to first hit this coexistence line, to first get phase separation, then of the wild type, then of the ARA minus variant. And it also explained why we never saw phase separation for the ARA minus minus, which was missing 2/3 of the aromatic residues, because its critical point is actually below freezing. All right, so, but there's more complexity, right? As we've heard a few times today. It became very clear looking at the composition of orthologs and paralogs of HNR and PA1, but also overall of Fed family RNA binding proteins, that they were very highly, that their composition was very highly conserved. And here we don't only have stickers and spacers in the sense that it seems important how much glycine and serine content there is, and perhaps how much charge content there is, because, or at least the high compositional similarity between these sequences suggested that. And so we asked the questions, are all stickers the same? What is the role of charged residues? And are all spacers the same? And I want to tell you about that. Looking at the stickers, Anna in my lab made a variant in which all aromatic residues was replaced by tyrosines, and one in which all aromatic residues were replaced by phenylalanines. And the all tyrosine variant had a stronger driving force for phase separation than the wild type and the all phenylalanine variant, as seen here by the lower saturation concentration. And we again used our trick of looking at single chain dimensions of these variants and of a bunch of other variants. And Mina and Rohit were able to extract interaction strengths for these different types of sticker-sticker interactions, as he also mentioned already earlier. All right, and this was, of course, in complete agreement with other people's work on aromatic residues and phase separation. Just, I think the advantage here is that we can actually put some numbers on it and really understand the strength of these microscopic interactions. Now, charged residues. Free and like low complexity domains generally have very few charged residues, but they seem to favor arginine residues over lysines, and they seem to favor a small positive net charge. So how do they overall influence phase separation? Well, that was not really clear from the literature, because depending on the sequence people were studying, it seemed like charged residues could favor phase separation via electrostatic interactions or cation pi interactions, or charged residues could increase the solubility and therefore decrease phase separation. And so this context dependence really made it hard to understand conceptually the role of charged residues in these sequences. And so to address this, we made a relatively large number of variants in which we titrated the number and type of charged residues to determine their phase behavior. You see that they make, in principle, nice condensates, except for this one sequence here, we're replacing all arginines with lysines. And indeed, when Anna in my lab replaced more and more arginines with lysines, this weakened phase separation until there was no phase separation visible anymore, really showing that lysine residues are highly solvated spacers. Then she looked at arginine residues and showed that when adding 7 arginine residues to the intrinsic 10 arginine residues of the wild type, it enhanced phase separation. It weakened phase separation, seen here as the higher saturation concentration, of course. So you needed more protein for phase separation. when removing 6 of the 10 arginine residues, it strengthened phase separation. And both of these data points would suggest that, arginines may also be highly solvated spacers. But then removing all of the 10 intrinsic arginines really weakened phase separation. And that showed, again, this context depends. It really depends on what's going on in the sequence, whether arginine worked as a sticker or as a spacer. We saw this context dependence also when looking at negatively charged residues, where adding more and more negatively charged residues weakened phase separation. But removing all 4 intrinsic aspartate residues also weakened phase separation. So can we understand this conceptually, or are we, do we have to look at every sequence independently to determine the influence of charged residues? So together with Rohit and Mina, we took inspiration from the solubility profile of globular proteins, which are, of course, least soluble at the pH that that is their isoelectric point, and become more soluble at different pHs because they have a net charge there. And we argued that the net charge per residue could be a similar parameter in for the driving force for phase separation of low complexity domain sequences if we kept the networking ability, so the stickers, the same in the sequences. And of course, we had this set of sequences where we only changed charged residues and didn't touch the aromatic residues, right? And when we plotted their saturation concentrations against the net charge per residue, well, we saw something that didn't really resemble this kind of V-shaped plot. So either this premise was wrong, or perhaps there was something additional going on. And these outliers would tell us something about perhaps we missed some stickers or some other effects in the sequences. So we made use of the analytical form of the stickers and spacers model that Rohit already introduced, where we can, we can estimate the saturation concentration from the number of stickers in the sequence. And there's a square here because, of course, you need 2 stickers to make a networking interaction. We said, okay, so if we have, in addition to aromatic residues as stickers, if arginine residues can also act as stickers, and we haven't taken that into account in our data or in the selection of our variants, then we can rescale the experimental saturation concentrations to take this into account, that our arginine residues can make interactions with aromatic residues. And when we did this and rescaled the experimental values, indeed, these data points for arginine variants are nicely moved onto this V-shaped plot. And then the lysine variants were still outliers. And these data points really suggested that lysines interfered with phase separation. And so adding to our model a 3-body interactions where lysine residues really interfered with aromatic and aromatic arginine interactions now nicely moved over all our data points onto this V-shaped plot. And to test whether this made sense or this was just by coincidence, we could fit this, right? We added a lot of other variants that were not used to fit these parameters, and they nicely fell onto this V-shaped plot. And so overall, this makes sense that we came to the conclusion, okay, aromatic residues are stickers. Arginine residues can act as additional stickers together with aromatics, and lysine residues really interfere with sticker-sticker interactions. And obviously, arginine and lysine have quite different properties. We have the guanidinium group and the arginine, where the charge is delocalized, and therefore the arginine is much less soluble than the lysine, where we have the point charge, and that really interferes with these cohesive aromatic interactions. We also really learned that the net charge per residue strongly modulates the driving force for phase separation via electrostatic effects, and hereby, 3 orders of magnitude. I also want to mention that the minimum of this plot here is not at 0, but at small positive values, which made it difficult to see these relationships in the data. And we really had to go through this analysis to see this. And in addition, the net charge per residue of the natural sequences also peaks at around that same value. And of course, we see a preference of arginines over lysines in prion-like low complexity domains, suggesting that the ability to undergo phase separation may very well be encoded in these sequences. All right, so what about the spacers? Because we said about 60% of the sequence is made up of just glycines and serines. Does it matter whether it's glycines or serines? Can we just change their relative ratio? It could be that there might be additional interactions through the hydroxyl and the side chain of the serines, and it behaves more like a weak sticker. Or this bulk in the side chain here could reduce the ability of the stickers to interact and could interfere with phase separation. And so, you know, this raises the term effective solvation volume. I think Rohit mentioned it. You know, we're thinking about how much volume does the side chain take up, either by its normal bulk or by how much it likes to be solvated and therefore expands the chain. So which of these properties is more important for the phase behavior of these sequences? To answer this, Anne and Wade in my lab made a number of variants where they replaced increasing numbers of glycines with serines, and this weakened phase separations. And when they replaced all serine residues with glycines, this strengthened phase separation, really arguing that serine is indeed a spacer, not a weak sticker, and glycine is the more favorable spacer of the two. We can play these same kinds of games also with other polar residues like asparagine to glutamine or serine to threonine, and we always see that the larger spacer will interfere with phase separation more. Oh, and there is actually the data. When we're going from serine to threonine or from asparagine to glutamine, we're weakening phase separation. Rohit also showed you that they can extract all of the interaction parameters from our single chain dimensions and can really simulate the phase behavior of our variants very well, even of variants that we haven't used in the parameterization. And overall, then we come to an understanding of how phase separation is encoded in the sequences of these prion-like low complexity domain. There is a networking component via the stickers, and we can understand what parts of the sequences, which residues are stickers, and their different interaction strength with each other. There's the particular lysine that really interferes with the sticker-sticker interactions. And then there is a density transition, and that is how well that is allowed depends on the effective solvation volume of from the spacers. And charged residues really like to be solvated and take up space, and residues with smaller side chains allow for sticker-sticker interactions very well. And then we have a mean field net charge per residue based term where a low net charge per residue leads to relatively low solubility of the sequences and allows for strong phase separation, and high net charge per residues values lead to electrostatic repulsion and solubilize these sequences. This also means that intrinsically disordered proteins can be highly soluble, right? I showed you only very few sequences that were highly soluble, but of course we're starting out with phase separating sequences. So just because a protein has a disordered region, it's not necessarily useful to assume that it will undergo phase separation. And with that, I want to show you one piece of data from an experiment from Eric Martin in my lab in this paper, where he used rapid mixing to look at how quickly phase separation happened, how quickly phase separation was nucleated. He started off with the HNRNPA1 low complexity domain in a solution without excess salt, where it's highly soluble, and then jumped through rapid mixing to a condition where he was in the two-phase regime, and then was able to follow how quickly the solution nucleated. And he followed this by small angle X-ray scattering, so that you can see small changes in the dimensions of the protein and as early as 60 microseconds after mixing. And what he observed was that the protein first compacted, and that reflected that in the salty solution, the protein became less soluble, and now we could see the effect of the aromatic interactions, of the sticker-sticker interactions, and then it started to assemble. While this mutant, which missed 2/3 of the aromatic residues, didn't really show any change. And so there's already a difference in the behavior after a fraction of a millisecond in this protein. This all points to work that Rohit's lab has done, and that he also talked about earlier. So I'll only point to it briefly, but connected with our data and say, this shows us that condensates are network fluids. There is a network of sticker-sticker interactions inside these condensates, and they're expected to have viscoelastic properties. And this density transition that is required to get to dense phases and coexisting dilute phases is mediated by spacers largely, as we saw in our case, lysines and serines, or large charged residues, and the net charge per residue of the proteins overall. And if there's no density transition, we could get higher order complexes, or at best at high concentration, a system spanning network. So we need to think about networking and about the density transition. And since these condensates are expected to be viscoelastic, at least on some time scales, perhaps it's not so useful to say liquid-liquid phase separation. Just talk about phase separation with some other properties. Of course, folded domains have an influence on the phase behavior, or can have an influence on the phase behavior of intrinsically disordered regions. I don't want to show you any data here, just want to point your thoughts in that direction. And where we looked at the influence of the two-folded domain on the phase behavior of the low complexity domain, and really came to the conclusion that, yes, we need to think about networking and solubility, but not only of the low complexity domains, but also of each of the individual domains and of their ability to make interactions with each other. And have we learned anything overall from these simple in vitro systems about the more complex multi-component stress granules in cells? I would say yes. And it's now become really clear that stress granules are phase-separated entities. I'll point you to these three papers by labs of Paul Taylor, Cliff Brengwin, and Simon Alberti. And they really came together to show that G3BP is the major stress granule hub that's important for the phase behavior, or for the assembly of stress granules, at least under redox conditions. And G3BP phase separates together with RNA. And if they make mutants of G3BP that change the phase behavior and alter how much RNA is needed, or abrogate phase behavior, and when they are introduced into cells that don't have any G3BP at all, and so can't phase separate, then the ability of these mutants to rescue the formation of stress granules is directly reflected by their in vitro phase behavior. Why would that be the case if stress granules have a lot of different components, and in vitro there's only 3 components, G3BP, RNA, and water? Why would that complex multi-component Rnp granule in cells, why would that be recapitulated by such a simple system? Well, the answer seems to be that G3BP is the central node of the network of protein-protein interactions. And it recruits all of the other components into the system. And this means now we have again a network fluid in cells, like the network fluid that we just talked about in vitro, just that now there is a network of different proteins and RNA, but we can think about this in very similar ways, that there is a physical network of different interactions and a density transition that leads to the formation of these kinds of condensates. And with that, I will close and say stickers and spacers can help us understand the phase behavior of proteins, whether that's the intrinsically disordered proteins that I talked about, or the linear multivalent proteins that we've also heard about today, or even folded proteins with patchy stickers and spacer regions in between. But in addition to stickers, the spacers are important, and one should consider the overall solubility. And with that, I'd like to close, and I apologize for the problems with sharing, and I'd be happy to answer questions. Thank you.
Thank you very much, Tanya. That was terrific. So we do have two questions already in the chat. Pritam Mukherjee asks, how does the presence of proline in a disordered region affect phase separation?
Yeah, that's an excellent question. Well, so what we know about prolines is, I think, comes a lot from elastin-like proteins. They, of course, have very different types of sequences than the prion-like low complexity domains. There don't tend to be a lot of prolines in the type of sequences that we've looked at, and so we have not investigated what proline would do in our types of sequences. In elastin type sequences, you have an LCST transition. So when you raise the temperature, you get phase separation, and that could be a combination of the fact that you have aliphatic hydrophobes, and overall this is an entropically driven phase separation, and perhaps the effect of prolines. I think there is more work that needs to be done to really know what the molecular basis for the LCST phase behavior is there, and how prolines affect it.
Thank you, Tanya. And Ben Schuster praises you with terrific work. Can you comment further on the role of asparagine versus glutamine, citing the fact that glutamine is 10% of the sequence? Sorry, asparagine is 10% of the sequence.
Yeah, so, different types of these sequences have different balance, right? In our case, we have more asparagines than glutamines. If we go to FUS, if I remember correctly, FUS has more glutamines than asparagines. It really changes also the fibrillization behavior of the proteins. But in our case, we know how we have studied how it affects the driving force for phase separation. So the asparagine has a smaller side chain than the glutamine. If we replace all the asparagines with glutamine, then we see a weakening of phase separation. So we have a higher saturation concentration and need a higher protein concentration to first see condensates. And that's in agreement with the glutamine having a larger side chain, and so interfering a little bit with the ability of stickers to interact with each other and to network.
Thank you. So with that, Tanya, I'll ask you to stop sharing, please.
Mm-hmm.
And I will now proceed to closing remarks. Institute for Quantitative Biomedicine crash courses would not be possible without the efforts of many individuals working behind the scenes. For their flawless execution today, my co-organizers and I thank Maria Voigt for designing and developing the flyer, Michelle Sangera for handling registration and Zoom logistics, Ken Dallenberg for IT support, and IQB Associate Director John Hackett for his leadership. We thank our speakers for their time and scholarly contributions, and for making this IQB crash course such a stimulating and intellectually rewarding learning experience. We thank Steve Labuti and our respective institutions, Rutgers, the State University of New Jersey, the Rutgers Cancer Institute of New Jersey, and Princeton University for their support. And finally, we thank you, the audience, for participating remotely. My IQB colleagues and I look forward to hosting you either in person or online for future crash courses. Please revisit the IQB webpage, iqb.rutgers.edu, in the late summer to learn about our fall semester crash course offerings. For the safety and well-being of all during the ongoing pandemic, I urge you to get vaccinated and boosted if you're medically eligible, and to take appropriate non-pharmacologic measures to prevent the spread of SARS-CoV-2 wherever and whenever they are warranted. As we close, Please join me in praying for a speedy and just end to Russia's war of aggression against Ukraine, and for all those who have lost loved ones, the thousands harmed physically and emotionally, and the millions displaced from their homes. Goodbye and be safe. Thank you very much.
Thank you, Stephen. Thanks, everyone.